Corinda sativum tried to reduce down the cholesterol to some extent. So these were the chemical parameters. Then if we talk for the oxidative stress parameters in testes, then come to the LPO, that is lipid peroxidation. In group 2, this LPO level was elevated, whereas the plant doses, they have tried to recover this LPO. Similar, if you see the antioxidant enzyme, that is the SOD level that was declined in the group 2 in comparison to that of the group 1, and the plant doses try to restore this antioxidant enzyme, which is very, very important for this mutation of this superoxide oxygen into the singlet oxygen. Then this catalase, another say antioxidant enzyme, this level was also declined in the group 2 in comparison to group 1, and the plant tried to restore this enzyme too also, because this enzyme actually catalyze or break down the H2O2, which that is the hydrogen peroxide free radical, into H2O and O2. So this enzyme was very much declined in the group 2, and this was recovered by the extract of aqueous and alcoholic of this plant. Then non-antioxidant, that is GSH. This was also, the level was very low in the lead nitrate treated group in comparison to the group 1. And the other aqueous and alcoholic, they again try to improve the level of this GSH. Then come to the testosterone and sperm density. Now, testosterone is the hormone which is produced by the testes, that is the cells of the testes, and it's very important. Now, the level of this hormone that was also declined when the animals treated with this lead. That means the heavy metal plays very important role. They may alter the level of the testosterone hormone. So the level declined, and this testosterone, not very, say, significantly, but try to bringing it to almost normal level, but not very much. It's not very much significant. If you see the sperm density is also very much low in the group 2, and the plant extract tried to, not significant, but try to normalize it. Now, what I told previously, that how do I come to know whether the lead is present in the testes or not? So that is checked by the AAS, that is atomic absorption spectrophotometer. And you can see in the graph that very elevated level of lead nitrate is seen in group 2, the animals which were treated with lead nitrate, and the group 3, 4, 5, 6, which were supplemented with the lead nitrate, the aqueous and ethanolic extract of the Corinda sativum, they tried to reduce the level of the lead. So this was the testicular parameters. Then comes the histopathology, that is histological. The figure 1 shows the normal architecture of the testes. Nothing, seminiferous tubules, everything, they are very normal, Sertoli cells. But in the group 2, the architecture, the normal structure of the testes, that was, I'm not telling that is damaged, but that is altered. This was seen with the lead nitrate. And here in the group, in the figure 3 and 4, you can see that to some extent, the recovery observed. Why I have conducted a histological, histopathological study in order to confirm the results which are obtained in the biochemical parameters. So that's what is observed. Then if we talk for the liver, then you can see the same, that AST, ALT, the enzymes, biochemical parameters, elevated in the group 2 and recovered in the other plant treated groups, in the ALT also. Then the ACP and ALP, in the group 2, you can see high elevated, significant, say, upregulation of the ACP and ALP observed, and downregulation of the ACP and ALP observed in the further plant-treated groups. Similarly, if you see the protein, then the protein is very much downregulated in the group 2 in comparison to group 1, and the plant tried to recover it. and the cholesterol level that was very much elevated in group 2, and the plant tried to restore towards the normal side. And what I observed, in some cases, the high dose is very effective and the low dose is not, but low dose is effective in almost all the parameters. And ethanolic extract and the aqueous extract, almost they are working together, maybe because ethanolic is slightly better in comparison to aqueous. Maybe the reason that the aqueous is more polar in comparison to that of the ethanolic, although that is also polar, but not very much polar in comparison to that of the water. Then comes the oxidative stress. You can see the LPO. This LPO in group 2, that was very much, say, increased in comparison to that of the normal animals, and the plant extract tried to restore it. Similarly, SOD decreased in the hepatic tissue in group 2 in comparison to the group 1, and all the low and high doses of this plant, that is coriander, try to normalize the SOD level. Similar is the case with the KIT. The level of KIT declined in group 2, and the plant doses try to, say, elevate the level of the KIT. Then if we talk for the glutathione, that is GSH, the GSH level declined in the group 2 in comparison to that of the group 1, and the plant doses try to restore it. Then if we talk for the histology, then you can see the figure 1, the liver lobules, they're normal in the treated animals. Hepatocytes, everything, they are clear. But in the figure 2, when mice treated with this lead nitrate, the distortion of the hepatocytes occur. This was observed. Here, the aqueous and alcoholic extract of this plant tried to restore the structure of the of the liver. Then come to the kidney, renal tissue. Here, the ALT level in the group 2, that was very much increased in comparison to group 1, and the plant doses again trying to normalize it. Similar is the case with the AST, that is elevated in group 2 in comparison to 1, and the plant is trying to, say, bringing towards the normal side. I'm not telling that completely it's restoring, because it was a crude extract, but let me tell you that the crude extract that is working more in comparison to go for an isolated compound, because here I've not taken the standard also, I've not taken any isolated compound also at that time, but now I'm working on many more things. Now this ACP level in the group 2, that is elevated in comparison to group 1, and the ALP level, that is also elevated in comparison to group 1, and the plant extract again normalizing it. Protein, if you see the protein, then they are the building materials of the body, and the level of protein is again very less in group 2 in comparison to group 1, and cholesterol that is increasing in the group 2 than the untreated animals, and the plant is trying its best to provide the therapy to the recovery to these animals. Then the oxidative stress parameters. The LPO level in the case of renal tissue, that was increased when the animals were treated with lead nitrate, and the SOD antioxidant enzyme, that is drastically reduced from the in comparison to that of the group 1, and the plant doses, again they were trying their best to provide the protection to the animals treated with the lead nitrate. Then this KIT, that again antioxidant enzyme, that is reduced in the group 2, and the glutathione, that is also reduced in comparison to group 1, but again the plant is providing its preventive role. Then if we come to the histology of the renal tissue, the figure 7 shows the normal structure of the kidney, whereas the figure 2, which is treated with the lead nitrate, here the distortion that is observed in the kidney. And these figures, they show that the plant is trying to provide the normal structure to the kidney. So now the final comes, and that is the discussion where I want to focus. See, in all the 3 tissues, that is testes, liver, and the kidney. In all the 3 tissues, I'll tell you. See, we have talked about the oxidative stress parameters. First, I'll talk that is LPO. Why this LPO level was enhanced in the group 2 animals treated with the lead nitrate? Actually, what happens? When the lead is given to the animal, then this lead induces the generation of reactive oxygen species, that is the free radicals. And these free radicals, they damage the cellular components, which are including, say, polyunsaturated fatty acid, say, species of phospholipid compounds. Now, what happened when the cellular damage takes place, then the level of the lipid is enhanced. As a result of which, what happens? The peroxy radicals are formed. And these peroxy radicals, they get rearranged and results in formation of a final product, and that is what the MDA or TBA, and that is the reason of elevating the level of the lipid. Now, when this was seen, I mean, in all testes, liver, and the kidney. Now, when this coriander sativum extract was given to the animal, and when this LPO level was to some extent normalized, what I found, why this so happened? Because this plant contains the antioxidant, and that is the flavonoids. And these flavonoids, they actually bind with the free radicals, that is reactive oxygen species. They bind with it and scavenge them from the body, try to eliminate them from the body. So once they are eliminated, then what happens? They don't get the chance to cause the damage to the cell membranes. And so the lipids, their level, that is not enhanced, and that is declined. This is what I observed. Then if we talk for the SOD, then SOD, what I said, that this is antioxidant enzyme, and this causes dismutation of the superoxide oxygen into singlet oxygen. And this SOD actually works in conjunction with another antioxidant enzyme, and that is catalase, which catalyze the H2O2 into H2O and O2. And both the enzymes, that is antioxidant enzyme, SOD and catalase, for their enzymatic activity, they require the various trace elements. Now here what happens for their enzymatic activity as well as for their molecular organization. Now when this lead is given to the animal, then what happens? This lead interacts with these various trace elements. And these antioxidant enzymes, they don't get these various elements. So when they don't get these various elements, what happens? The level of these enzymes, this goes down. And when the extract of this coriander sativum was given to the animal, what is found? That again, I'll say that it contains what? The flavonoids, antioxidants. And these flavonoids, directly, they are not, I mean, elevating the level of SOD or the catalase. I'm not telling this thing. But these flavonoids, they have a tendency that they may bind with the ROS or the free radicals which are generated by the lead, and then try to eliminate them from the body. So once they are eliminated, maybe by a minor extent or a major extent, and results in what? Elevation of these antioxidant enzymes. Then if we talk for GSH, that is non-antioxidant, the lead is having a high affinity because this contains the thiol disulfide group, that is GSH. Now, this lead is having affinity for this thiol disulfide, and so its level goes down. But when this is given, this extract is given, it contains such ingredients that it elevates the level of this GSH. This was seen in all the tissues. Then, if we talk for the biochemical, if I say for this, what do you call it, the AST-LT, then both these enzymes, they're linked with the, say, carbohydrate metabolism, they're linked with the, say, amino acid metabolism. And when the lead enters, this lead actually causes the cell lysis. When the cell lysis occurs, what happens? These enzymes, they are released into the blood circulation and result in the elevation of what?
अभी दिखाई दे रहा हूँ? मैं अभी खाना स्टार्ट करूंगा, अभी खाया नहीं है, रुक गया मैं.
I'm audible now? Hello?
Yes, ma'am, audible.
Yeah. So when this plant was given, it was found that this plant possessed very active ingredients like polyphenols, as what Dr. Navneet Sharma was talking. So these polyphenols, they play a very important role in preventing the lysis of the cells. And if we talk for the ACP, then ACP actually, when lead enters, it causes the assimilation of the ACP and thus increases the ACP activity. And when the plant was given, and this may be due to like lysosomal imbalance. So when the plant extract was given, this plant extract tried to normalize that lysosome so that an enzyme should not come out. So this was the reason. And further, like we talk about this sperm density or what called testosterone, then already what I said, that the lead treatment highly, having the impact on these hormone and the sperm densities, but the plant extract that tried to restore it. I said that the tasty structure was not damaged, that was altered. So the normal architecture was to a greater extent provided by this plant extract so that the normal level of the testosterone can be achieved. So this is what I have talked about, the biochemical and oxidative stress and histological parameters. And in all the tissues I have seen that the lead concentration was elevated by the AAS and the plant has tried to restore it. Then finally, if I come to the conclusion, then I'll say only single sentence, as I didn't know much. The plant is it's a Corinda sativum. I say that it's a traditional, say, medicine, and it's an edible vegetable, possess, say, very active bioactive compounds, active ingredients, having polyphenols, having, say, coumarine, isocoumarine, corindrine, having glucosides, having, say, phytic acid, having phthalate. Even phytic acid is acting as a methylchelating agent. So that chelates and try to remove this toxicity from the body. Later on, I have tried by taking the standard 2 also. I've isolated the compounds 2 also. I have done the NMR, FTIR, LCMS, possible structure of the novel compound have been elucidated. That I have not presented here. Because what I said, keeping in mind the students of PG and the young talents, research students, I've taken a very preliminary and a basic study. So in the end, I would say that from the bottom of my heart, I thank the entire team of the conference organizing team, and I'll take, because I was regularly in contact with Dr. Siddharth and Dr. Bhavna Paliwal. They were very polite, very soft. Whenever I called, promptly they respond me. If I've not taken the name of anybody, please apologize me. I'm thankful to all the organizing team. And one more thing, I want to thank the Department of Bioscience Biotechnology, this Vanasti Vidyapeeth, who actually provided the facility for performing this type of experiments. Then one thing, foremost and last but not at the least, I would like to thank the Almighty God, because without His grace and blessings, I stand nowhere. And very important, and that is what I would like to thank my research students, Richa Shukla, Tania Agarwal, Sakshi, and I think the other students, they are not here, Anjali and Surbhi and Kuso. They were very helpful to me in preparation of these slides, in spite of their busy schedule, because I'm also busy in my this examination. So if I'm forgetting anything, so again, I'll tell you, thank you very much for inviting me and providing me this platform to present this very basic talk. Thank you very much.
Thank you so much, ma'am. It's a very lovely and very awesome, wonderful, fruitful, informative talk. I am very thankful to you. You have come in this session. And I hope now any question answer from audience. audience asked any questions?
I think it's a very basic work. Everyone understands. I don't know very well everything.
Yes, ma'am.
I thought that in a very brief time, I mean, keeping in mind the young talents, I've chosen such a study so that they may understand it.
Yes, yes, ma'am. Miss Bhavna Paliwal, I hope you are listening me. Miss Bhavna Paliwal.
Hello.
Yes.
Yes, Siddharth sir.
Say something about ma'am and conclude this session.
Ma'am, first of all, thank you very, very much that you just got your precious time to us and was this much involved in this conference. And this is actually very sweet. The things you have said for me and Siddharth. You are also that polite person we have, what being such down to earth. She's been working so hard. I had seen her personally. I had like learned from her, and I know that अगर कोई मैम को लिसन कर रहा है हाँ ढंग से तो उस बंदे ने कुछ लाइक उनका दिमाग खुल ही गया होगा. She has that knowledge that if she's saying something she'll be delivering it with everything she has. That's her nature, and ma'am, you're incredible.
Bhavna,
Yes,
I'm
ma'am.
so thankful. I'm so thankful to Siddharth. Again, I'm so thankful to the entire team. I mean, tireless job you all are doing. और बहुत बहुत धन्यवाद. बहुत अच्छा आपका. मैंने सुना जो लेक्चर्स बच्चों के भी सुने और मैंने कुछ डेलीगेट्स से और जो रिसोर्स पर्सन्स थे उनके भी मैंने सुने और मेरे को बहुत अच्छा लगा कि मैंने ज्वाइन किया और मेरे को कुछ सीखने को भी मिला. मेरे को बहुत सी चीजें आई लर्न फ्रॉम इवन फ्रॉम द स्टूडेंट्स टू ऑल्सो. मैं तो हमेशा बोलती हूँ कि मैं बच्चों से भी सीखती हूँ कुछ बहुत सी चीजें. तो आप लोगों से मेरे को बहुत कुछ सीखने को भी मिला. आई एम सो थैंकफुल टू यू.
थैंक यू मैम. एक्चुअली मैम भावना का काफी बार मैसेज भी आया कि आई जस्ट मिस्ड बनस्थली विद्यापीठ. मैं सोचा कि चलो एक बार अभी बात ही कर लो.
हाँ हाँ बिल्कुल. आप आइए. आप आइए. आप आइए बनस्थली विद्यापीठ आइए. आपका बहुत स्वागत है.
जी
हम भी आएंगे आप तब मिलेंगे definitely.
Okay, now conclude this session and I hope as per schedule we can extend our program.
Just a minute, Mr. Siddharth. Mr. Siddharth,
yes.
Just a minute. Yeah. Thank you very much, ma'am. This is Sushil, Dr. Sushil is speaking. Yeah, it was a mind-blowing lecture session. It was very much interactive, and there are several participants from the Banasthali Vidyapith. So the entire credit goes to the Siddharth and the team working behind this scene from the Banasthali also to making this entire event a grand success. In that, you have made it excellent by your prestigious presence, outstanding sharing of knowledge and experiences. Your experiences and knowledge boon us and give some incitement in us also to work upon the fields related as you have delivered the topic. And I also talk to you. Meanwhile, you are very much busy in the administrative duties and several academic duties of the university because it's an examination session going on over there. So I can understand, ma'am. You have given a lot of time. So we are very much grateful and appreciative for your kind presence over there. Thank you, ma'am. Thank you so much.
सर मैं आपको नमस्कार करती हूँ. मैंने पहले अपना क्षमा मांगी थी कि अगर मैं कोई नाम नहीं ले पा रही हूँ. आप सभी का बहुत बहुत धन्यवाद. मेरे को ध्यान है कि एक दिन पहले मेरे पास फोन आया था रात को. तो उस समय मैंने कहा कि मेरा थोड़ा सा इस समय एग्जाम चल रहा है, ड्यूटी वगैरह है, मैं मतलब कल ज्वाइन नहीं कर पाऊंगी. सो मैं आप सभी का बहुत बहुत अपने तहे दिल से आप सब लोग का धन्यवाद देना चाहती हूँ कि आप सब लोगों ने मेरे को ऐसा मौका दिया कि मैं अपना कुछ थोड़ा सा काम आपको बता सकूं. सो आपको बहुत बहुत धन्यवाद. थैंक यू वेरी मच.
it will be a pleasure for us whenever the session and the time allows. We will absolutely meet to you at the Banasthali Vidyapeeth for the future assignments and
Sure.
consultants as well. Thank
You're
you.
most
Thank you so
welcome.
much.
Most welcome. Should I leave, Siddharth?
Yes, ma'am. Yes, ma'am. Sure, ma'am.
I have some work also. Okay.
Okay. Sure, ma'am. Sure, ma'am.
Okay.
Okay. Thank you, ma'am. Thank you.
Thank you, ma'am.
Okay. Nice.
Now our next guest speaker from National Institute of Biologicals, Dr. Avid Ali Sheikh, sir. Dr. Avid Ali Sheikh, sir. May you hear me?
Hello.
Yes, sir.
Yeah, am I audible?
Yes, sir. Very much audible, sir. Very much.
Okay. So should I start?
Sir, just a minute. I am just introducing you.
Okay.
From other participants. Dr. Abid Ali Sheikh sir is now serving as a training official scientific as National Institute of Biologicals, Noida, Uttar Pradesh, India. He served as research scientist from 2017 to 2019 at Seri, Kashmir Institute of Medical Science, SKIMS Kashmir, Jammu Kashmir, India. He teached a biotechnology discipline at Government Degree College, Department of Higher Education, Jammu Kashmir from 2015 to 2016 in academic session. Earlier, he worked as assistant research scientist in pharmaceutical biology Axial Biomed Private Limited, Hyderabad from 2007 to 2009. Dr. Avid Ali sir has got expertise in research as well as teaching in the field of his specialization. It is pertinent to mention here that he has many international publications to his credit and presented paper in international conference synopsia. He is member of many international scientific organization and editorial board member of various journals. Now I would like to warmly welcome Dr. Avid Ali Sheikh sir. Please sir, and I will also request you to Ms. Bhavna, please share the PPT of Dr. Avid Ali Sheikh sir.
Okay.
Please, sir.
A very good afternoon to all. The organizing committee, Mr. Siddharth, and all the team members who are associated with this very prestigious, important scientific event being organized from 12th to 14th, 2022. So I am highly grateful to you all for inviting me to have a simple, I can say, very brief conversation about the new regime of therapeutic models, molecules in the field of biological science, which have been recently developed by the scientific innovations and revolutions, what we call biorevolution. So I will be talking on the biological or biosimilars as the scientific biorevolution innovations for the human health and the betterment of the society. Is it audible, Mr. Siddharth?
Yes, sir. Yes, sir. Very much audible. Please.
Okay. So, first of all, what are basically the biological molecule what we are concerned about? There are different organizations which are governing the medical science or the health of the human population is concerned. The premier and the worldwide organization which we know is World Health Organization, WHO. So as per the WHO's recommendation and the terminology is concerned, biologicals or the biological products are those components or the entities which are derived from the cells, the tissues, or the microorganisms, which are living bodies having the inherent variability or characteristics of living material characterized in their genomic or the hereditary entity. and at the same time they are very much active substances so far as the efficacy or the activity is concerned, with having a complex structure originated from the biological source. They are of complex nature with diverse characteristics in their chemical biology or their interaction nature is concerned, with having a different from the source as well as the target is concerned. And at the same time, when it comes to the US based Food and Drug Administration, FDA, which is governing authority as per as the approval for the drugs or other cosmetics or the food products which are to be consumed or which are to be used in the human health as well as the other purposes of the consumption and other related affairs.
Water
As
crisis
per,
not a new issue for India and the world in the present context. We come across various articles highlighting.
hello, hello, hello.
Sir, please unmute yourself. Sir, please.
I have no.
Yes, yes, yes.
Okay, okay. It was sorry. Sometimes like the mic gets.
Sir, no,
Okay.
actually, I am here unable. Okay, okay,
Yeah,
sir,
yeah.
please.
Oh, sorry for the inconvenience. So far as the FDA's recommendations are concerned, like biological products or those medicinal components or the medicinal agents which are made from a variety of natural source. It could be a microbe, an animal, or a human cell, or any biological entity or a living creation who could be used as a source for the production, generation, or as its component could be used for the medicinal purpose. This is regarding as far as the FDA's statement or its recommendations are concerned. Like drugs, also they have normal drugs which are in clinical practice now earlier, chemical agents or those natural products. Like them, those biological agents also have they are produced with this intention of treatment for or the prevention of different diseases as well as the diagnostics for various chronic disorders or the genetic or epigenetic issues or problems are concerned, so that predispose of those disorders are concerned, we can diagnose at an earlier stage those problems. So biological medicine, so far as the European Association is concerned, biological medicines are those which are made by or derived from the biological sources as such, which could be bacteria or yeast, because most of the earlier as well as the current those drug molecules are the agents which are used as a large scale. They are you produced from such simple sources or such biological simple biological agents which are having an exponential lifespan of production property of multiplication property and easy source for their production and their commercialization is concerned. They could be explored and they are used for the production of those biological agents, and they are related consist of small as well as the complex components with various features and diverse characterizations at their each component or the periphery is concerned. Now, since biological agents differ from chemical entities in various characteristic features, the most prominent is their structural characterization or their overall this interactional behavior is concerned. They are produced from a living source with the inherent property of specialization and post-translational modifications and other characteristic features which are normally not found in case of the chemical entities or the synthetic drugs or the generic drugs which are produced in uh, by chemical methods or in vitro synthetic approaches. or from the natural sources, like we can say the plant molecules or the other antioxidants with a normal structure which is reproducible and having the similar properties as their parent component is concerned. But this is not the case in case of like biologicals or biosimilars, which have a different pattern of synthesis, biosynthesis, as well as the different pattern of interaction of activity is concerned. So on an average, if we see the biological agent has got a huge cost for its preparation as well as its maintenance. So on a daily dose of biological agent cost around 22 times more than that of a simple chemical drug for any therapeutic regime of disease is concerned. So it is very costly because it is production as well as the maintenance, and it is like we can say the development is concerned, it is very tedious, complex, and time consuming, as well as needs high infrastructural and high skilled behavior, as well as the manpower so far as its production or its uh, commercialization is concerned. So that's why like this therapeutic regime is not too much highly explored across the world or adopted still now. But there are different patterns or different platforms where scientific community is working to minimize its costs by producing the replica in the form we will be talking in future, that are produced the carbon copy of like those particular bioactive agents. Those are known as biosimilars. We will talk in the in next few slides about those. So in I can say like biologicals are the large complex heterogeneous mixtures of uh, living entity or the molecules or the biomolecules which have genetically engineered cells or the normal uh, process is concerned, their production. So after particular transcription and translation process, they have got modifications and their post-translational changes, which may result in different type diverse activities, relationships, or targets, or their affinity is concerned for a particular surface ligand. So it is highly versatile as well as highly focused process where we need the expertization as well as the accuracy of the production is concerned, so that the target molecule or the target cell or the target gene is properly produced, created, generated, or we can say is properly maintained so that it can achieve the target. Because having a high cost, high manpower, or high infrastructure requirement, so its skills or its standard of production is highly cumbersome as compared to the normal traditional chemical drugs. So there is a difference between normal chemical drugs as far as with respect to the biological and chemical drug components are concerned. Those molecules are relatively small with low molecular weight, simple, defined, and independent of manufacturing process. Like they could be produced individually in so far as their target or their production is concerned for a particular type of drug molecule or the parent molecule. For a chemical synthetic method is concerned, they can be independently or separately produced in any commercial scale production unit for without any like other component, or without the combination of the second drug is required, or some complex serious sequential experimental procedures are required. So well defined structural elucidation is already predefined. The target based synthetic approaches are used for the chemical synthesis of those components, and at the same time the chemical processes can generate identical copies of this parent molecule with a number of like independent number. We can generate enough copies or enough molecules at the production commercial level for the same particular drug molecule without any so much of complex hindrances. and it is easy to characterize the complete sequence of events or the chemical reactions are concerned in a particular production process far as chemical drug synthesis is concerned. So it is a predefined, synthetic method of production of there is scale of events involved. So they are already pre-designed or characterized with the standard as far as the synthetic chemical methods are concerned. And the component produced can be remain stable for a period of time as far as its stability is concerned with a predefined temperature, moisture, and other uh, conditions are concerned, though it could be maintained for longer duration for use of the health. So at the same time, the chemical drugs are normally non-immunogenic too much because their production or their rate of reactivity is concerned. They are normally chemical in nature and they react with the target cells in such a way or the target so far as their particular production is concerned. They have been already produced for some specific purpose. And as compared to this, the biological agents are mostly complex and mixture with a huge molecular weight and have heterogeneous structure or heterogeneous manufacturing processes required because their structure may involve some like post-translational modifications, some uh, additional characteristic components to be added so far as the target based approach is concerned. And in this approach, we have to include some more bioactive agents to the components so that we can go for a particular target based therapy. In this regard, the process of production is complex, heterogeneous in nature, and the biological agent itself has been produced from a genetic code with its variability. So we have to maintain the production or the generation conditions in such a way so that the biological process or the uh, generation of this bioactive molecule is not having any post-translational or other issues with the actual design which is actually targeted or aimed for the production of this biomolecule. And at the same time, those chemical agents are not normally uh, when there is some extraordinary compound which is allowed to get patented, but normally so far as the biological agents are concerned, they are protected by patented rights of the production agency. So their regeneration or their copywriting is prohibited until and when the uh, patent expires, then only its replica could be generated by using the biosimilar technology. Completely, we can't characterize the composition because of having a complex nature and also involving too much of high throughput technology, but because slowly the biological and technological scientific innovations are coming with the new methods and the new uh, instrumentation which facilitates the resolution of the bio molecules at very minute level so that the interaction or the efficiency or the characterization of those three-dimensional structures are resoluted at a very base level so that we can characterize the complex process, but complete characterization of this whole mechanism is not being yet achieved as far as the biologicals are concerned as compared to the chemical agents. And they are highly, as I said, they are highly sensitive towards the condition where they are maintained or where they exist or where their production is maintained, because being a natural compound or a natural cell or a natural biomolecule in, so they can be highly sensitive for those conditions, so that so definitely biosimilars, as I said, they are produced because normal generic. हेलो हेलो
Yes, sir.
हाँ आवाज आ रही है
जी सर बिल्कुल बिल्कुल
हाँ so innovation increase in innovation technology has enabled for the biological field for the market respective to the most expensive of the treatment options, and global consensus on the need to reduce the price of treatment has resulted in the development of certain products which are of the same pattern and having the same type of efficacy as the biological agents. They are known as biosimilars, but they can be produced only after the ex expire of the patent for the particular biological agent is concerned. Then their copy can be produced, which is known as a biosimilar. Biosimilar is exactly the same type of uh, compound, or the I mean sorry, biological agent with which is produced in the natural environment of a cell or a tissue, having almost the same efficacy as far as the its parent biological agent is concerned. But the difference is like their uh, production cannot be uh, or started until the expiry of the parent patent of this it is brand biological agent is not over. When the patent for the biological agent expires, then only the industry or the scientific community or the manufacturers can develop a similar type of biological agent, which is called as a biosimilar for that particular brand biological agent. So there are three main uh, types of biological therapy regimes currently in progress or in the pipeline, we can say. One is the biologics or the biological agents or the biologicals simply. They have been developed 2 decades ago. They are in the leading phase. They are under some are under trial, some are being already approved by FDA for treatment process, and so some are being developed and again used in the treatment regime. So this is which last 2 decades has shown a very much exponential increase in the field of biologicals. But in the developed nations, because they have a high health standard, they can afford such treatments in the because the companies which are producing those biologicals are mostly of them are in the Europe or America based those organizations. So at the same time, like having a novel therapeutic target based approach originating from the biotech processes or the biological processes itself natural, these are patentable. As I said, those components are the biological agents. Once produced, they are patentable. They are have a reference price already fixed or proposed from different organizations of the world for governing or regulating the health uh, parameters. And now, so far as the biosimilars are concerned, they are similar to the organic or the parent biological agent with competitive bioequivalence in its activity or efficacy is concerned. They are not patentable because they are produced for the commercial purpose by a pharma company just a replica of the parent patentable biological agent and generally produced with a lower price. They are intended for the common health problems of a common health man, so far as which may not be able to afford the biological high cost therapy for their treatment options. Biosimilars produce or provide a alternative, achievable, and possible solution for those patients. Now, combining the biological and biosimilar treatment options, a new domain of therapeutic modality have developed, which are known as biobetters. A biobetter is superior to its originator. Chemical or molecular modifications are done for its efficacy, activity, and lower its side effects and to maintain its cost at a achievable rate and enhance pharmacokinetics and pharmacodynamics. But having a higher price, again, those better components or therapeutic molecules have got uh, limiting limiting factor for its achievable or accessible process is concerned. Then we can say like every therapeutic molecule, once it is developed, manufactured, commercialized, and consumed for its purpose, there are different regulating authorities across the world. Some are national, some are international. So far as our country is concerned, we have our own a central board for regulating those drug and cosmetic acts. So far as in India it is concerned, like importing, manufacturing or sale or distribution of drugs is concerned, we have a drugs and cosmetics authority which were produced different rules and regulations. We have drugs and cosmetics act 1940 and 45 which us describe and show us like how to maintain and regulate different components or the biological agents with different activities concerned, their regulation, their consumption, their sale, and their manufacturing process are concerned. So these acts and their regulations act, have a check and account, make ensure that the safety of the consumers, the public, and the health standards are maintained in a standardized manner, and the quality of the products or the imported drugs are maintained and standardized for the common public in a standardized manner. In this similar way, like Chemical Drugs Acts in 1940 and 45, for biologicals and biosimilar products, the regulatory pathway, manufacturing process, safety, efficacy, and quality aspects for biologicals and some biosimilars, pre-market regulatory requirements are there definite for post-market analysis are concerned. They have been already predefined and designed by the central agencies for those biological and biosimilar agents. They are, in case of biological and biosimilars, the acts are a bit different, but they have not changed or replaced the 1940 or 1945 regulations, which are for the chemical drugs. But in addition to that, these regulations have specifically been developed for the biologicals and the biosimilar agents for particular treatment options. So in this regard, we have Indian Pharmacopoeia official standard for those regulations and for standardization of those drugs and other nutraceutical products and biological agents for which are manufactured, marketed, and which are being imported in India for the treat and used for the treatment of prevailing diseases which are currently in affecting most of the population in our country. For this purpose, the Indian Pharmacopoeia has established a complete official book of standards which has been prepared, developed for the standard of the treatment regime and also for the operation of different companies and other regulators which offer health benefits to the society and also to the manufacturing units for ensuring the health standard. Next. So, so far as the career options are concerned, like so far as the career options are concerned in biological or other this research oriented field is concerned, there are different career options for hello.
Yes,
Hello.
sir. Yes, sir. Yes, sir.
Okay. So far as the career opportunities in this area is concerned, we can have different like career options in this field. Like we can have R&D sector, we have production section, we have quality assurance, quality control, and we can have the regulatory domain of it and the small material vigilance and other opportunities where the students or the researchers can develop and progress and excel for achieving their targets, and they can be involved for in this study. At the same time, like uh, as we discussed about biological biosimilars also have different biological. हेलो
जी सर.
आप ये दिखाइए कंक्लूजन, ये इसमें एडिशनल स्लाइड है अभी ये प्रेजेंट में, इसमें हमारे कुछ अपने 2-4 स्लाइड है, ये तो उसके लिए रखते हैं. आप नीचे 2 स्लाइड.
Okay. Bhavna, please shift the slides.
आप दूसरा दिखाइए.
Last 2
slides.
or last 3. Yes.
हाँ. तो basic difference between those clinical advanced biosimilars and those the biological agents. We should have, we can develop and use the biosimilars as compared, because they are the need of our for the common public's health interests are concerned, because they are having almost the same efficacy, same target oriented achievements, but of at a lower price, and without any patent or that they can be commercialized, and the companies are now shifting towards the domain for production of the biosimilars replica of the biological agents after their patent expires. So many types of cancers today are curable at early stage, like here breast here two positive breast cancer by trastuzumab. This is the monoclonal uh, antibody drug developed as a biosimilar developed for the treatment of breast cancer HER2 positive patients. It has been developed as a biosimilar agent based on its parent monoclonal antibody biological agent. And in the similar way, quality of life has enhanced with the monoclonal antibodies like adalimumab in rheumatoid arthritis. And at the same time, the relapse of the disease has been reduced with the rituximab in non-Hodgkin lymphoma, and better patient compliance for example, blood transfusion in CED was welcomed by the erythropoietin and Daraviton. Widen treatment options for no drug, less options therapy in many diseases for you so far as the biosimilars are concerned. Now, market advantages, like these are the clinical advantages obviously over the other drug components, or it is master class of molecules, the biologicals. Biosimilars have clinical efficacy comparable to those, but at the same time, the market advantages are it is very much interesting, like affordability. The common public can manage since the drugs for the chronic diseases like especially cancer, diabetes, cardiovascular or neuro disorders, they are of high cost and they are produced mm, from highly expertise and established manufacturing units, which are normally like biological agents or immunotherapy is concerned, the hormone therapy or the gene therapy is concerned. In case of some neuro disorders or genetic disorders, they are normally produced outside India in developed countries, Europe or America or other places. So their import is very much costly, having a high huge tax and other production market rates. So normal patient common man may not be able to afford those medicines, and the patients ultimately suffer and they have a miserable end with the way the problem or the disease progresses, and they are not able to get even a single dose of immunotherapy or hormone therapy or the gene therapy or the stem cell therapy. So for that purpose, once those standard references are available and their patentable time has expired, the companies or the pharmaceutical organizations are now focusing on the biosimilars production, and they are really having almost the same efficacy and activity parameters, and at the same time at an affordable rate the common patient can manage or have some sources to get it be available at developing nations or at the normal institutions or the health sector infrastructure as well. And at the same time, opportunity for the biosimilar companies has opened a window for those marketing as well as the manufacturing units to get those uh, compounds and those active substances to be developed at a commercial level. So at the end, I would uh, conclude by saying like biological agents have potential with a promising results. They are of high interest now, especially in case of oncology sector. This domain is concerned. They have highly uh, productive and positive results with different types of cancers being therapeutically treated as well as diagnosed at an early stage using the biological agents or components or the cells or the genes, and also better response with fewer side effects. If they have respond positively, the patient and the host's immune system have a positive compatible response with the biological agent as far as the other regime is concerned. But with hello, so
Yes, sir.
we can use those biological agents as a supplement with our chemotherapy, because normally chemotherapeutic drugs have been standardized for most of the cancers at earlier stage. So since the biological therapy regime is recently developed in near 2 decades, so since the established treatment options are surgery and the chemotherapy and radiation therapy. Currently, many of these drugs are produced and tested in clinical trials, but the problem is that they take years being tested in clinical trials to gain their approval by the FDA as their trial process, and since they are new, so obviously, and it has a biological characteristics and it has to be included in a biological living system, so definitely there are different barriers and different parameters which takes time for its characterization and validation, and ultimately then its approval by the FDA takes too much time for being uh, launched in the market as a therapeutic agent or as a candidate for uh, clinical practice. Okay.
Okay, thank you so much, sir. It's a very informative talks. Yes, Bhavna, please.
Actually, I was not able to talk in a normal way. I talked very low tune because I was sitting in the office area. I was not able to go to my room or
Okay,
some.
okay, sir. Not an issue, sir.
That's
Not
why
an issue, sir.
I talked in a low tune and in a confined short way.
Okay,
That's
okay,
So
sir.
still you managed to deliver such a brilliant presentation on renovations, and we that was very fruitful and informative for us. And thank you that you got your precious time and presented in front of us. Thank you so much, sir.
okay. Thank you very much.
Thank you so much, sir. And now, as per our schedule, now the time of the presentation time. And after the presentation, we can listen our next guest speaker, that is from Saloni University, Dr. Devjyoti Bose, sir. So now, as per schedule, we can start our presentations from the invite delegates and the participant of YSO and YSP. And our next participant is Mr. Mayank Raj.
Yes, sir. Hello, sir.
You can present your paper.
Yes, sir.
Yeah. After that, next participant is Mukesh Kumar AR. Then this is the last participants, and then start will presentation of Khalsa
Visible,
College
sir.
Patiala. So be ready after that.
Sir, my screen is visible, sir.
No. Your screen is not visible. If anyone want to present your paper, then please contact me as soon as possible. Who cannot present last 2 days or 3 days?
Sir, screen visible, sir.
Yes, very much visible. Please proceed.
Yes, sir. Thank you. Good afternoon, everyone. Myself, Mayank Raj, from Maharishi Markandeswar deemed to be University, Mulana. I am pursuing PhD under Dr. Manoj Singh, Associate Professor of Department of Biotech. Today, I am going to present my research topic, biomimetic approach for synthesis of silver nanoparticles and characterization from aloe vera aqueous extracts. Nanoparticle. A nanoparticle is a small particle that range 1 to 100 nanometer in size. Nano derived from the Greek words nano, which means dwarf, yeah, extremely small. A nanoparticle interfacial layer is a well-structured layer of typically organic molecules, ions, etc. Properties of nanoparticles. Nanoparticles often possess unexpected optical properties as they are small enough to confine electrons and produce quantum effects. Nanoparticles of yellow, gold and gray silicon are red in color, and gold nanoparticles melt at much lower temperature. Absorption of solar radiation is much higher in material composed of nanoparticle. Then it is in thin films, continuous sheets of materials. Why silver used in silver nanoparticles? Silver is one of the basic elements. Silver nanoparticles are increasing various fields: medical, foods, healthcare, consumer, and industrial purpose and chemical properties. They have been used in several applications: antibacterial agents, industrial, household, healthcare, consumer products, medical devices, optical sensor, cosmetic products, pharmaceutical industry, food industry, etc. Silver nanoparticle used for the surgical, prosthetics, and fungicides, etc. What is silver nanoparticles? Silver nanoparticles size 10 to 100 nanometer, chemical formula Ag. Silver nanoparticles are ultra-fine particles of silver. They are 10 to 100 nanometer large and differ from the bulk silver as they have different colors such as yellow as opposed to the silver. I may choose to aloe vera plants. What aloe vera? Aloe vera is a cactus plant. Aloe vera contains phytochemicals, polymannans, atracunins, mannans, and thorn glycosides such as imodine and various lactins compounds are present in aloe vera leaves. It is used in many consumer products: skin beverage, ointments, gel, form of the minor burns, sunburns. It is help to wound healing effect. Green synthesis of nanoparticles. Green synthesis of nanoparticles using plant constituents like carbohydrates, fats, enzyme, terpenoid, polyphenol alkaloids, and reducing agents to synthesize silver nanoparticles. The use of plants, microbes, and fungi in the production of silver nanoparticles is leading the way to more environmentally and eco-friendly. Advantage of green synthesis of silver nanoparticles: eco-friendly, low cost, can be used for large scale synthesis, no need to high pressure, energy, temperature, and toxic chemicals, not required any special culture preparation and isolation techniques. Application of green synthesis of silver nanoparticles: it's drug delivery, cancer treatment, DNA analysis and gene therapy, antimicrobial agents, biosensors, etc. They have two objectives: synthesis of silver nanoparticles and characterization of silver nanoparticles. First of all, I am discuss synthesis of silver nanoparticles. Use material and hello sir. Hello.
Please.
Sir, voice audible, sir?
Yes, very much audible.
Okay, sir. Material methods: aloe vera plant was collected to MMDU campus Mulana. Collected sample for preparing the aqueous solution. I have cut small pieces in aloe vera leaves in small sizes in beaker 200 ml. Into boiled, cut a small piece, boiled to water bath at 50 degrees Celsius. Sorry, 60 degrees Celsius for 5 minutes, and filter the Waterman filter paper grade 1 to store 4 degrees Celsius to next use. To remove non-silver nanoparticles components along with maximal recovery, supernatant and periods to recovered synthesized solution and optimal centrifuge process, bare centrifugal for 10,000 rpm to 14,000 rpm, time duration 15 minutes to 20 minutes by purification process. Synthesis of silver nanoparticles: I have cut a small piece to dissolve in distilled water and filter the aloe vera extract to Waterman filter paper and mix with AgNO3 silver nitrate to mix and change the color of dark brown to the change in the colorization of mixture. We have made indicates the synthesis of nanoparticles, AV silver nanoparticles, and for the confirmation we will proceed with differentiation characterization techniques. I have 4 types of characterization are used in UV visual spectroscopy, DLS dynamic light scattering, and TEM transmission electron microscope, and EDS energy dispersive spectroscopy. And next I have discussed first UV visual spectroscopy. UV visual spectroscopy is fast, easy, simple, sensitive, selective for different types of nanoparticles, need only a short period of time. Aging silver nanoparticles have unique optical properties which make them strongly interact with specific wavelengths of light. It was observed that maximum absorption is 4 nanometer. It is different time interval 0 minute, 1 hour, 2 hours, 4 hours, and 5 hours. 5 different time intervals. The DLS is a method that depends on the interaction of light with particles. This method can be used for the measurement of narrow particle size distribution, especially in the range of 2 to 500 nanometer. DLS is non-destructive method used to obtain the average diameter of nanoparticles. And next, TEM, transmission electron microscope. TEM is a valuable, frequent, used, and important techniques for the characterization of nanomaterials. TEM shows the 10 and 50 nanometer size spherical and spheres present aloe vera silver nanoparticles. The magnification of TEM is mainly determine the ratio of the distance between the objective lens and the specimen and the distance with objective lens and image plane. EDS, energy dispersive X-ray spectroscopy, one of the chemical analysis system. TEM is energy dispersive X-ray spectroscopy was applied to quantify the chemical composition of the specimen. It shows the presence of a strong silver signal along with weak oxygen, carbon, and copper peaks. These signals are the fingerprints of the elements in the sample and can be used as a source of quantitative chemical and electrical structure information. Discussion point: the spectra show a well-defined surface plasmon band for plant between 400 nanometer, which is the characteristic of silver nanoparticles and clearly indicates the formation of nanoparticle solution. In the case of silver ion reduction of aloe vera occurred at 400 nanometer. The size of nanoparticles was found to be the DLS in the range of 20 to 110.1 nanometer. The peak number and peak amplitude obtained gave important explanation for size and size distribution of silver nanoparticles.
Okay. Please.
Okay, sir. The mixture of flat, spherical, and sphere shape obtained to TEM to range 20 to 50 nanometer size. The high resolution TEM image shows the particle were highly polydispersed in nature. EDS, it shows the presence of a strong silver signal along with weak oxygen, carbon, and copper peaks, which may have originated from the biomolecules bound to surface of silver nanoparticles. Conclusion slides. Nanocomposites of silver nitrates were synthesized by using the plant extracts of aloe vera plants. Nanoparticles are synthesized under eco-friendly conditions with less toxic chemical production. Nanoparticles are then characterized by using different analytical techniques such as UV visual spectroscopy, DLS, TEM, and EDX, and many types. Same FTI are used. Finally, if we succeed in all these studies, it would help the research of the nanoscience and nanotechnology community to develop safer, biocompatible, efficient cancer or anti-angiogenic agents containing silver nanoparticles. Eventually, to ensure the biosafety of use of silver nanoparticles in humans, studies dealing with biocompatibility of silver nanoparticles and their interaction with cells and tissue are inevitable. Finally, the great concern is that the developing nanotechnology-based therapy should be better than available technology, and it should overcome the limitation of existing treatment technique. Finally, it has to provide a safe, reliable, and viable treatment of disease with high accuracy in patient-friendly manner. This is reference page, and some articles get published in this year. Thank you, sir, for listening.
Thank you so much. I have some inquiry.
Yes,
Before DLS, TAM, or UV, how you
yes,
can confirm कि your particle is AZNP or not?
sir.
I have
जैसे
you can only 3 characterization, right? DLS, UV, and
no, sir,
TAM.
4 characters.
And
Yes, so
next
UV,
one is
DLS, EDS, sir, energy dispersive spectroscopy, sir.
अच्छा, तो you are just characterized TAM EDS or SAM EDX?
Sir, TAM EDS.
TAM EDX. Why you cannot check characterized by FTIR or XRD?
Sir,
This is the primary. I hope this is the primary characterization, right?
yes, sir. It's because sir, I am the research in MSc dissertation. Plus,
Okay.
XRD is not available in this institute.
And TAM EDX are
Sir,
available
TAM,
in MMDU?
no, sir. TAM EDX nahi. This sample is, I think, Punjab University. तो sample वहीं से तो result आए थे sir. TAM and EDS से.
Okay. Thank you so much.
Thank you,
Now
sir.
the next participant is Tamanna Devi.
Hello, good afternoon. Am I audible?
Yes, you are very much audible. Please proceed.
Okay, just a minute.
I hope this is the after this presentation we can listen our guest speaker Dr. Devajyoti Bose from Ceylon University. After that, other participants are joined. Please, ma'am, proceed.
Okay, sir, is my slides, are my slides visible?
Yes, very much visible.
Okay. So, good afternoon, everyone. Myself, Tamanna. I am pursuing my PhD in Maharishi Markandeshwar Deemed to be University under the supervision of Dr. Manoj Singh, sir. He is an associate professor in the same deemed university. Today, I get the opportunity to present my knowledge on the topic. My research topic is isolation of bacteria from poultry litter and its future perspective when used as manure. Just a minute, sir. Okay, so starting from the introduction, talking about poultry, global poultry production is massive in scale with tens of billions of chickens, turkeys, ducks, and other poultry species raised every year for meat and egg production. The last few years has seen an impressive research effort devoted towards the developing and validating methodologies for poultry welfare assessment and management methods related to it. Poultry litter is the waste created in the biggest amount all the while. It is generally the waste considering excrement, feathers, stage of the feed, support metrics, which can differ as per the nation and the locality. For example, the sawdust, shavings, nutshell, rice husk, and others. The litter also contains various plant nutrients such as nitrogen, phosphorus, potassium, trace elements such as copper, zinc, pesticide residues, pharmaceuticals such as coccidiostats and endocrine disruptors, etc. However, besides its organic content, poultry litter can be contaminated with various types of pathogens, including viruses, bacteria, parasites, and fungi, so that the microbial concentrations in chicken litter can reach up to 10 raised to the power 10 CFU per gram. So there are the three main objectives of my research. First one is to isolate different bacteria from poultry litter. To identify the isolated bacterial species from poultry litter is the second one. And the third one is to study the future perspective related to poultry litter used as manure. So the materials and methods of the research somewhere goes like that. The chemicals and various media used were procured from high media, Aldrich, Merck, and Sigma. The chemicals and various reagents were used, and the media used during the research work were nutrient broth, Mueller-Hinton agar, and McConaughey. Various instruments were used, and various equipments and glasswares were used for the project. Now, talking about the methodology. Now, starting from the sampling, the collection of samples were done during the period from February 2022 to April. I collected 40 poultry litter samples randomly from the different areas of the sheds of hatchery located nearby. Directly transferred in the sterile Ziploc packets to the laboratory for further preparation and examination. Second step followed was the preparation of sample suspensions. 1 gram of poultry litter sample is weighed and transferred into a sterile flask containing 9 ml of nutrient broth. The content of the flask was homogenized, and then the suspension was incubated at room temperature for 24 hours. Later, the suspension was cultured MHA and McConaughey agar for 24 hours. Then the colonies were isolated and they were identified. Bacteria isolated was identified using Maldito. Talking about the results, on Mueller-Hinton agar, we observed creamish white elevated colonies, while on the McConaughey agar, there were some red pinkish colonies and some were whitish brown colored colonies. Actually, on McConaughey agar, lactose fermenting bacteria, for example, E. coli or Enterobacter other species or Klebsiella species, produce acid by utilizing the lactose in the medium, resulting in the appearance of red or pink colored colonies, while that the bacteria that do not ferment the lactose, such as Salmonella species, Proteus species, etc., will instead use peptone, and this produces ammonia, which raises the pH of the agar and causes the plate to form white, colorless, or golden brownish colonies. So this gives an indication that on our plates we get both kind of colonies. So means the bacteria we isolated were of both the kinds, lactose fermenting as well as there were some colonies of non-lactose fermenting bacteria as well. After that, we have done the Maldito of MS. As shown in the table, we got various results. There were different kind of bacteria that were isolated from the poultry litter. The poultry litter samples taken from the hatchery sheds were found to be highly contaminated with several pathogenic and non-pathogenic bacterial species, including E. coli to be the maximum, that is 55%, Comamonas species, we obtained 12.5%, and Enterococcus faecium 5%. There were also some other bacterial species, for example, Bacillus pumilus, Citrobacter coceri, Citrobacter sedleci, Proteus mirabilis, and Campylobacter jejuni having 2.5% share. Now the discussion. E. coli as a ubiquitous commensal germ present in human and animal intestinal flora. So it was obvious to have E. coli in our results. Enterococci species are also one of the fecal contamination markers. So they can be found in the chicken litter as well. Bacillus subtilis and other species are the bacteria found in fresh litter generally, but can also be a part of old litter. They are usually used as the prebiotic-based cleaning products in the chickens, so we can obtain them in their litter as well. Now, coming to the conclusion part, the findings show that E. coli is the most overwhelming bacterial species to be identified from the poultry litter. This may be because it is an inhabitant of gut of animals as well as humans. Various other bacterial species which were isolated and identified by Malditov, such as Enterococcus species, Proteus, and Campylobacter species, are the pathogenic bacteria that cause several diseases in poultry birds. In contact through any means. Proteus species are generally found in poultry litter, as we have discussed earlier, but can be found in older litter as well. On the other hand, the bacteria we have found, such as Comamonas species and Citrobacter species, there are not proper investigations done on the basis of their role in the field of poultry or subjects related to poultry. Now, talking about the future perspective of chicken litter as an manure. Chicken on land as an organic is the cheapest and most environmentally safe method, according to the previous studies. Um, but little is known about the safety of chicken litter for land application and general release into the environment. Bridging this knowledge gap is crucial for maximizing the benefits of chicken litter as an organized.
नहीं, ये तो फिर मैं क्या शायद कोई दूसरा सेशन में घुस गया, वैसा है क्या? इन दैट केस मेरे को लिंक एक बार फिर से भेज दो ना, दैट वुड बी बेटर देन.
Am I audible, sir? Okay, so I was talking about the future perspective of chicken litter when used as a manure. Actually, the key safety concerns of chicken litter are its contamination with pathogens, including various bacteria, fungi, helminthes, parasites, protozoa, and viruses, as well as antibiotics and antibiotic resistant genes. Antimicrobial resistance is global multifactorial problem that involves animal, food, environmental, human sectors. The poultry sector is among the fastest growing agro-based industries. So to increase the yield, they use various kind of feeds and antibiotics. So these antibiotics through the food chain also enter the food chain of humans. For example, as shown in this figure, various antibiotics are used by the broilers and poultry farm owners, and they injected these antibiotics to the birds also through their feed or any other means. When this chicken litter is used as manure in the fields, so from the manures, it reaches to the soil, to the water, and to the plants, which were consumed by humans in the future. So these antibiotic resistance transmission route enters the food chain and affects the humans as well. But not proper research is yet done on this topic. So it opens up. It opens the huge chance or opportunity to help further research on this topic. Thank you.
Thank you so much, ma'am. Nice presentation. And now the next presentation by Kamalpreet Kaur from Patiala College. Kamalpreet Kaur, Akashveer Singh, may you hear me? Anyone present from Patiala College? Hello. Hello. May I audible? Hello, may I audible?
Siddharth, you're audible.
Okay. Dr. Devjyoti Bose sir. And Dr. Devajyoti Bose sir, can you hear me? Sir, please unmute yourself. Anyone from Khalsa College, Patiala? Hello. Amandeep sir.
Good afternoon, sir.
Hello.
Sir, in case if Kamalpreet is not speaking, I'm Dilpreet here. Can I begin with my presentation?
I think yeah, I will just ask the moderator to just check on that.
Our next guest speaker and plenary speakers are joined with us, Dr. Devjyoti Bose sir from Saloni University. Dr. Devjyoti Bose sir is a associate professor of biotechnology at Saloni University. He completed his graduation in chemical engineering from the West Bengal University of Technology and post-graduation in renewable energy with specialization in bioenergy with PhD in chemical engineering on microbial fuel cells for wastewater treatment from the University of Petroleum and Energy Studies, Dehradun. He is a member of the American Chemical Society and Society for Chemical Industry and is an active researcher in the area of bioenergy. He has 40 plus research publications to his credit in well reputed international journals with several book chapters and conference publications. His experience in the field has afforded him the opportunity to become familiar with various kinds of waste to energy system, especially in microbial fuel cells, biomass, pyrolysis and water bioremediation bioreactors such as sediment MFC, double chambered PT catalyzed MFC and second generation biomass derived activated carbon air cathode MFC are some of the systems developed in the research project as part of extracting energy in the form of bioelectricity from influents. While reducing the contamination level of such wastes, his work on microbial fuel cell was covered in the news section of Science last fortnight. Quarterly Journal of Indian Academy of Science in 2018 and received the Best Researcher Award from UPES Dehradun for conducting bioenergy research during the same timeline. He possesses a combined teaching and research experience of over 4 years at UG-PG levels. Now I would like to invite warmly welcome Dr. Devajyoti Bose sir. Please sir.
Yeah, so thank you for that introduction. Am I audible?
Yes sir, very much audible sir.
Okay, and you're able to see my screen?
Yes, very much.
Okay, great. Thank you so much for that introduction. Right, you've really covered almost everything. So a big thank you for that and also for inviting me to this session at recent trends in life science and biotechnology. So what I'm going to do is I'm going to cover an aspect that is very deeply linked with everyone. Okay, that is refineries. Okay, and that waste materials that you get from there and how we can use microbes to maybe look into that. Because remember, suppose your automotives, you're using diesel engine, petrol engine, it comes from the refineries. Cooking oil, LPG, your gas cylinder, that also comes from the refinery, right? Then a lot of other things like naphtha, with a lot of things used in the polymer industry, the paint industry, everything comes from oil refineries. So what I'm going to do is I'm going to talk about the waste which poses a problem to the human health from there and how we can use microbes for that. So I think The session expert has already given a brief background on me, and so my area is sort of a cross between chemical engineering and biochemical engineering, and we sort of have a lot of articles to our credit in that aspect. And we also have international collaborations from institutes in Canada and the United States, right? And also some IITs in India as well. And we work on this field of bioremediation, waste to energy, bioenergy, and all these kind of things. Okay, so that is just a brief about my profile, as you can see. So we'll get straight to the matter here, which we were discussing. And if you look at the right side of your screen, you'll find three circles. So everything starts with water, right? So we use water in everywhere, from cooling to sort of a lot of other processes. And now when we go for the treatment of this water, we consume a lot of energy, right? So which is the second circle. And this energy generation process that you have in whatever form eventually leads to climate change because of the emissions. And climate change, as you know, continues to disrupt water cycles here on Earth, right? So this forms a very vicious circle, which sort of, is the reason for the pretty much detrimental effect you have on the environment. And on the left side of your screen, you'll see a graph projecting India's emission problem in the next 10 years. So if you look at a year like 2032 or 2030, you'll see we're going to produce 3 to 4 billion tons of CO2 over and above what we are producing right now, if we don't do a reversal of our current trends, right? So let me give you an example. If you look at an average human being, okay, we consume about 2000 calories a day in terms of food. So if you convert that into electrical units, that is good enough to sort of, light about 6 light bulbs. 600-watt light bulbs can be lit by that. And so you might argue that how much energy do we need? Right, so we find that if you look at one light bulb, which consumes about 100 watt, that's about 25 watt down the drain if you convert the calories into the electrical unit. So we waste a lot of energy, what I'm trying to say. And if you look at the 25 watt, you can easily have a smaller CFL type light bulb, which can pretty much give you the same intensity lighting that you want, right? So that's the idea. This is how we optimize. Okay, we look at what is waste and we try to make the waste have the same impact as the source. So this can be done with the help of microbes. And in systems that we do this, these are called BES, bioelectrochemical systems. So this is from a multitude of disciplines, from fermentation to electrochemistry, to material science, to microbiology, to chemical engineering, to environmental science. Everything is crossed, okay, in this field. And what we do is we use microbes, okay, because they are a repository that is in abundance across the globe. They consume waste matter, right? So if you look at maybe microbes in the soil, you have Shigella and Geobacter. Okay, and so it's a combination of fermentation and respiration process, which we call bioremediation. Okay, and this sort of bioremediation is very, very effective when it comes to sort of cleaning waste. Okay, and so studies have shown that n number of waste can be sort of cleaned. So what happens is bacteria consume this waste matter and they release electrons. So these electrons can be captured by an electrical circuit. Okay, and we can produce electricity from it. So now coming back to the refinery problem, which I told you in the beginning, that oil refineries are unavoidable. They are everywhere, from your engines to your to cooking in your kitchen. Everywhere you use a product of the refinery. Okay, and this production process releases a lot of hazardous chemicals like polyaromatic hydrocarbons, okay, and phenolic substances, which are barely degradable by nature. And the process that we use to treat these material, because you cannot just dump them outside, right? Um, the chemical process itself consumes a lot of energy, and the entire process results in production of material. Those are carcinogenic, that means can cause cancer, which are teratogenic, which means can cause fetal abnormalities, as well as mutagenic, that is which can cause cellular mutations. And of course, the EPA, or the Environmental Protection Agency, takes a very grim look at this. So there is a need to find alternative to treat material that sort of comes out of an oil refinery. Okay, and if you look at sort of the waste that you generate, so a refinery essentially has three outputs. Okay, all are called distillates because they come from a distillation column. The heavy distillates, there are three distillates: heavy, middle, and light. So the heavy distillates are kerosene, industrial oil, heating oil. The middle distillates are jet fuel, diesel, light oil, and the light distillates are petrol, LPG, and the naphtha. Okay, so which eventually goes into manufacture of lot of solvents used by a lot of other industries. So refineries are an unavoidable fact of our societies, and we do need them. But the issue that comes is that we need to find a way to treat the waste better. And the issues with the existing system is they in no way can reduce the carbon emissions. Okay, we just can't reduce them. Okay, and the treatment mechanism that also you have now are very, very expensive. Okay, so this has led to the idea of looking for alternatives. Okay, so this is the point where your this bioelectrochemical system comes in, or PES as we call them, where we use microbes to break down petroleum or hydrocarbon contaminants. This field where you are using microbes to break down hydrocarbons, we call this field petroleum microbiology. Okay, so microbiology is the study of cellular mechanisms of a microbe. So apply that to petroleum. It's called petroleum microbiology. Okay, so we use microbes that can metabolize petroleum products, waste more to be precise. Um, they can biotransform, biodegrade, and bioremediate hydrocarbons. Okay, so these hydrocarbons are a broad, therefore, target of metabolism and also act as a substrate for the microbe to sort of break down. Right. Now, in terms of abilities, they can be sort of put into bioreactors. And if you put them in bioreactors, there are a lot of bioreactors that are available. So if you put the waste material into a system which can generate electricity from it, we call them microbial fuel cell. Okay. If you want to generate hydrogen from it, we call it microbial electrolysis cell or MEC. If you want to desalinate, say water particularly, we use MDCs, again using microbes, and then you have microbial electrosynthesis or MES, which is used to extract valuable minerals from the waste streams. So all these are an overview. Of course, I'm not going into too much detail because each will cover its own aspect. So these are some of the microbial systems that you have to generate, in some cases, electricity from the waste, in some cases, value-added minerals from the waste, in some cases for desalination as well. Okay, so these methods of treatment, we call them biochemical treatment, and they produce bioelectricity, they remove contamination, they show microbes that are adaptable to a wide range of pollutants as substrate. Okay, and reactions happen at ambient conditions, so you don't need specific conditions or hi-fi conditions. Okay, and the electrodes that you use are non-fouling in nature. That means the bacteria biofilm, suppose, that is forming generally doesn't tend to foul the electrode. Okay, it gets renewed every time. So at the end of the day, you have come up with something much better. So these are some of the systems you can see on the right hand side from our lab where we have used this waste material, and you can see one LED light being lit. Okay, so no plug point, no nothing. Directly from waste, you're generating electricity, and that electricity is used in an electrical circuit, okay, to produce light, okay, in a small LED, right? And the only thing responsible for this are the bacteria or the microbes present in this waste. So now obviously the idea comes that how these bioelectrochemical systems can be used in a petroleum refinery. Okay, so if we look at the Indian scenario, we generate around 28,000 tons of waste from the refinery processes every year. Okay, and globally it's said that about 1 billion tons of waste are generated. Okay, and the conventional method I said already are carcinogenic, teratogenic, mutagenic materials are released, not very efficient, not very energy, consumes a lot of energy, and doesn't really meet any environmental regulation. Okay, so, there's a lot of waste that is generated from this field. And so people started putting them in a microbial fuel cell and see how it works. So you have, if you look at the left hand side, you have an anode and you have a cathode. So the anode is where the microbial biofilm grows, and it consumes the organic matter, releases electrons, which travels the length of the circuit, and the reaction is consumed or rather completed at the cathode. And in the cathode, you have a triphase reaction of the electron, proton, and the oxygen. And of course, you have heterotrophs or other microbes growing on the cathode, which assist in the sort of the entire process. So studies have shown, so I've given you a timeline that people started investigating refinery waste matter from 2010, and they found 41% removal is possible of certain waste. Then another study 4 years later showed that not only waste removal is possible, even power generation from that waste is possible. Okay, and then finally one study showed high amount of power generation, close to 0.7 amperes, is possible from the system. So obviously, this has progressed significantly because remember, again, it's a biochemical process. So you don't expect the values to be very high, but these studies demonstrated that it is a possibility to use these hazardous waste material and get significant output from them in terms of reducing the toxicity of the material, in terms of producing electricity from them. So this is an ideal example of waste to works, and this is needed because remember, refineries are not going anywhere. Pretty much everything you do one way or the other is linked with a product that comes from the oil refinery. So there is no escaping that. So we need to develop these systems. So further people even developed more. So again, another timeline I've given you. Here you can see that from 2014 to 2019, a 5-year timeline of how they develop systems. And on average, people have been able to demonstrate that with certain cultures, you can have about close to 80-85% waste removal. So this is significantly exciting if you put it in research perspective, because we do want to remove the waste. We don't want the waste to remain that level of toxicity, right? And also we are able to generate a little bit amount of electricity from it, close to 350 milliwatts. Okay, so about 0.3 watts we can generate from these systems. So obviously, this is at present in purely the research phase, but the research phase has given us some really good answers. So we expect the technology to develop in the sort of the next few years. So it can be concluded that the refinery hazard material or wastewater treatment in the BESS can be effectively done and sort of can be well adapted for such kind of systems. Right. So what are the issues? The issues is huge quantities of waste are generated on daily basis. So that sort of, we need to find a way to handle that. Okay. And the refinery operations might get complicated because of certain, chemical flux into the microbial biofilm. So that needs to be checked. Then the application area has to be checked, like power generation, you want to use an MFC, desalination, you can use an MDC, recovery, you use an MEC. So a lot of aspects are there. Okay, and everything is interrelated from biotransformation to bioremediation. All these things are connected together. And of course, it will require eventually a pilot scale testing. At present, people are doing it in independent studies in batch bioreactors. Okay, of course, things like low economics will at present affect the issue, but these are things we expect science and technology to take up and eventually find the answers. So we look at a situation eventually where we'll be hopefully somewhere in the future, somewhere maybe in the next 10 to 15 years, we'll be able to integrate refinery processes with microbial processes like this. Okay, so you have the catalytic cracking process where the hydrocarbon that you have extracted from below the earth is cracked into the different products. Okay, the different distillates, the higher distillate, the middle and the lower distillates. And of course, after everything is done, waste from each of the processes can directly go into a microbial bioreactor or a BES, bioelectrochemical system, where we can treat the waste, we can generate some electricity from it, and therefore make the process much more sustainable than what it is today. Right. So this is some of the aspect. These are some of the references, and thank you. I hope that I've been able to give you some perspective about how this process works. And so that is from my side, the presentation. So thank you.
Thank you so much, sir.
Yeah, thank you.
I would like to invite you, Dr. Sushil Kumar Upadhyay, sir. Please say something about Dr. Devajyoti Bose, sir.
Yeah, thank you very much, sir. Very short notice you have accepted our invitation and request and given your valuable time to present this innovative idea, which is giving an scope to the academician in the industry-oriented aspects. Our curricula always try to design in such a way in which the amalgamation of the academics along with the industry perspectives. And this kind of research which you have presented here in front of the young scholars will incite the works in the refinery industries along with the microbial amalgamation. It may be an integrated approach of the microbiology, biotechnology, biochemistry, physicochemical science. All the things will definitely boost. And the microbial application in the refinery with respect to the VS will be a future for the young scholars also as a criteria of research. So I wish to request to all the young scholars which are very much interested in the microbial science, industrial biology, industrial microbiology, they must be tried to go through the methodologies and the agenda discussed by the Professor Devjit Koser. And it is a pleasure for me to listen several new and keen aspects in that as well. I listened the entire session very keenly, but the things that were mentioned in the form of challenges and then approaches to the integration with the microbiology was outstanding in my personal view. So it was an excellent, sir, and we are very much grateful to you. You have taken our consideration in a very short notice and given a valuable time to us and shared your experiences and best wishes also. So thank you so much, sir. Thank you very much.
Thank you, sir, so much for your kind words. Really means a lot. And again, thank you so much for inviting me. And again, I will look forward to similar interactions in the future also. So thank you from my side as well. Thank you.
Over to Siddharth, please.
Thank you so much, sir. Now, as per our schedule, we again start our presentation session. Now only 8 students are left for presentation of oral and posters. I would like to invite Dr. Amandeep Singh, sir. Please present your students from your side.
Hello.
Yes, sir. Yes, sir.
Thank you, sir, for providing the opportunity. First of all, I would like to introduce my first student, Dilpreet Kaur, and she will present its presentation.
Yes, sir. Sir, please on your camera and share your PPT.
Okay. Students need to share camera on, sir?
No. Just for interaction session.
Okay, sir.
Good afternoon, everyone. This is Dilpreet here, student of BSc Biotechnology, Khalsa College, Patiala. And I'm obliged to be a part of this international conference. And I'm honored to present my presentation to you all. My topic for today's presentation is psychological stress as risk factors for cardiovascular diseases. Sir, is my presentation visible?
Yes, very much visible. Please present.
Okay, sir. So we'll be discussing about stress and its common sources and how it leads to heart attack. After that, we'll see about acute and chronic stress and how they all are related with cardiovascular diseases, followed up by broken heart syndrome and atherosclerosis. So what exactly is stress? When you are in a situation where you fail to respond to a particular challenge, be it in terms of behavior, psychological or physiological, stress is always have a negative impact on you and can lead you to death. So how it causes cardiovascular diseases? When you are stressed, your heart rate will be increased, right? Your work rate of that heart will also be increased, causing that strain on your heart, making it to prone to diseases. Next up, we have the common sources of stress. Now, stress can arise from external and internal causes also. For example, if you are facing any major life changes in your life, right, your financial problems or your work, or when you're not satisfied with your work, the perfectionism that you want to achieve, but due to some reasons you are unable to. So these are the like sources of stress that how a person can be stressful in life. it's peer and social pressures, or when there are work situations. So these are the common sources of stress. How psychosocial stress is related to cardiovascular diseases is when a person is stressed, it is directly linked with its behavior, and which will eventually give rise to the risk factors of cardiovascular diseases. Next up, we have that how stress leads to heart attack. So when you are stressed, there's this region in your body, the region in the brain, that is amygdala, that basically sends signals to your bone marrow so that it can produce more white blood cells. Now, when there are more white blood cells, what they will do is they will inflame the arteries. There will be inflammation in the arteries, which will directly lead the heart to heart attack. Now, since amygdala is also known as the fear center of the brain, and when you feel anxious or stressed, it directly switches on and sends the stress hormones to your body to activate that fight or flight response. What it do is it basically reduces the blood flow, and it makes your heart deprived of oxygen and nutrients. So when you are in a stressful environment, what happens is you experience an oxidative stress, which will eventually cause the inflammation in your arteries that are directly linked with the cardiovascular diseases. That includes your hypertension, your heart failure, arteriosclerosis, myocardial infarction, stroke, and many more. So next up, we have acute stress. What exactly acute stress is? It is a situation that occurs instantly. Like, for example, when you are driving a car and you're about to hit by a truck, right? So there you have experienced a nearly death experience. That is acute situation. Or when you have scolded by a boss, right, for not doing your work properly, that is an acute stress. So what happens in acute stress is the hormones that are involved are epinephrine, cortisol, and norepinephrine. So what happens exactly is, as you can see in this picture, there is this coronary artery that supplies blood and oxygen to your heart muscles. So when these stress hormones are released, what they do is they basically inflame the arteries. They basically block the arteries in order to prevent the flow of the nutrients and oxygens to your heart, right? So as you can see is there are blood clots, and there is plaque buildup in the artery, which eventually depriving your heart of oxygen and nutrients. And how it works is in acute stress, there is release, or you can say the increased secretion of epinephrine and norepinephrine from sympathoadrenal, and cortisol from hypothalamic pituitary adrenal. What they do is they basically increase the heart rate and blood pressure, which eventually leads to your cardiovascular diseases. So as you can see here is the fight or flight acute stress response. What happens is when you are stressed, or when a person is stressed, the stress hormones will be released, right? There will be heart rate, there will be increase in heart rate, and your heart will pump in a like very efficient way, and it will be deeper breathing. There will be increased blood flow to large muscle groups, and the responses, the reaction to this is that there will be increased ability to handle stress, right? Your body and mind will be prepared to overcome that situation, but in the same way there will be inflammation in that arteries, right? There will be increased inflammation, and it will deprive your heart to receive those oxygen and nutrients. Next up, we have chronic stress. Chronic stress, as we have already discussed, that acute stress is basically a situation where a person is experiencing stress on an instantly, like instantly he is experiencing stress. Whereas chronic stress is a stress where your emotion gets accumulated in your body for a longer period of time, right? And as a result, you experienced high blood pressure and heart diseases. Next up, we have broken heart syndrome. This broken heart syndrome, also known as stress-induced cardiomyopathy or Takotsubo cardiomyopathy, which is a condition that arises when a person is stressed, right? And it happens. It usually causes sudden chest pain, and when there is increased rate of release of stress hormones such as adrenaline that basically damages your heart. So the complications related with broken heart syndrome are low blood pressure, pulmonary edema, heart failure, and blood clots in the heart. As you can see in this picture, there's a normal heart and there's a heart that is a broken heart syndrome, that shows broken heart syndrome, right? You can see in the normal heart that is in a normal shape. like after contractions and all, it is completely in a normal shape. While Takotsubo cardiomyopathy or broken heart syndrome, as you can see, is the left ventricle is bit swollen up. So what happens here is the heart is in irregular shape when a person experiences stress. Next, we can see that when a person is stressed, right, due to fear and there will be fear and behavioral changes due to stress, and those stress hormones will be released from the brain, right, and those neurotransmitters will releases those stress hormones, which will eventually cause pain in your entire body, but specifically in your heart also, and there will be muscle pain as well, which as a result will lead to your cardiovascular diseases, be it heart attack or stroke or angina, right? And next we have the treatments and drugs. So to treat such cardiovascular diseases, we have these kinds of treatments and drugs that can be followed up. First of all, we have psychotherapy. What exactly psychotherapy is? It is basically a situation when you attend some sessions with a person who is in stress or who experience anxiety. These are the normal sessions that are conducted by a psychologist, right? When you're stressed or when you don't know what to do in a particular situation, when you're anxious, you try to visit your psychologist, and they try to give you suggestions, or they try to guide you in a better way. Next up, we have medicines. The medicines to treat such diseases can be angiotensin 2 receptor blockers, right, or beta blockers. Next, we have complementary approaches. Now, in complementary approaches, these are the alternates. For example, if you're stressed, it is always advised you to do your like when you're interested in something, to do that particular thing, right? For example, if you're interested or you are like habitual to sing, so try to sing or try to dance. So these are the complementary approaches that can be done to control your stress. Do yoga or do exercise, eat properly. So these are the complementary approaches that can be used. Next up, we have benzodiazepines. These are basically to treat stress and anxiety. The examples for this are alprazolam and diazepam. They basically helps to send calming signals to your brain in order to stop that overthinking. So when you are stressed, you can go with the benzodiazepines for in order to so that your body or your brain can send calming signals to reduce the symptoms of anxiety. Next up, we have beta blockers. What they do is they basically they basically decreases your blood pressure by blocking the effect of adrenaline. Adrenaline is what? It is a stress hormone, right? So in order to block that effect, the beta blockers can be used. And next up, we have antidepressants, for example, selective serotonin reuptake inhibitors, which can be used in order to cure or in order to maintain that stressful conditions. So next up, we have the mechanism of atherosclerosis. Atherosclerosis is a condition where there's a buildup of fats and or the other substances in your artery walls. What happens in atherosclerosis is when there's a release of stress hormones in your body, it will eventually make your arteries narrow and cause the buildup of plaque and fats in your arteries, right? Because due to the release of stress hormones, your arteries are blocked and your heart is deprived of oxygen and the important nutrients that requires to make it functional. Cortisol is responsible basically for this atherosclerosis because when you are stressed, it is released and it floods the bloodstream with glucose and it narrows your arteries. So it basically happens when you are prone to high blood pressure or high levels of glucose, fat, and cholesterol. Now what happens exactly is when stress hormones increases, it will eventually increase the deposition of fats and cholesterol in your arteries, triggering your arteries to be inflamed due to rise in blood sugar levels. So as you can see here is, it is presented over here that there's an artery, right? And due to the stress, when stress hormones are released, there's the buildup of these plaque and fats and lipids all over it, so that it prevents, or it is basically depriving your heart to receive those nutrients and oxygen that it requires to be functional. And as a conclusion, we can see that emotional stress may act acutely to trigger major cardiac events. Now, since stress is one of the major factors leading to cardiovascular these days, Many other like complementary approaches can be taken in order to reduce your stress, so that you don't like prone to cardiovascular diseases. You need to improve your diet. You need to visit your psychologist on daily basis if you're experiencing stress in your daily life. And well-known phenomena of stress-induced cardiomyopathy seem to support that the belief that severe acute psychological stress can precipitate cardiovascular diseases. Thank you so much. Other references that I have taken. Thank you.
Okay, thank you so much, Dilpreet. Next one. Next participant from Patiala College.
Good afternoon to one and all present here. It is an honor to have the opportunity to address such a distinguished audience. So let me introduce myself. My name is Mannat Aliwal and I am a student of biotechnology at Khalsa College, Patiala. As you can see, my today's topic is applications of biosensors in food industry. As you can see, these are the contents which are displayed on my screen, which we are going to discuss today. To begin with, food industry is basically emerging as a prominent industry nowadays, and it is associated with the production, processing, storage, handling and monitoring of food. And we all know that food is vital for life. Now, with the increasing thrive in this industry, there come out certain parameters which are of major concern, like monitoring and maintenance of food quality, purity, and its nutritional value. This can be technologically assessed by biosensors. The author of biosensor is Dr. L. Clark, and this is a typical biosensor here. So what is a biosensor? It is basically an analytical device which contains an immobilized layer of biological material which is directly in contact with the transducer. The transducer here is a sensor which senses any kind of biological activity and then it converts to electrical signal. This electrical signal is amplified and then it is recorded in a micro equipment which is also again electrical, and then this signal can be recorded and can be studied further. This is the schematic representation of the biosensor, and the principle on which it works is signal transduction, which we have already discussed. That is conversion of biological signal into electrical signal. So this leads us directly to my next point, that is food biosensors. Now, food biosensors are of two types. First is enzyme biosensor, which finds its use in beverage and liquor industry for analyzing carbohydrates, amino acids, etc. The second type of biosensor is used for spotting any kind of microorganisms in food, be it good or bad. The significant or prominent features of biosensors, because of which they are used in food industry, are electronic integration, stability, target specificity, mass producible, continuous signal, rapid response time, and their small size, which make them portable. Now, I would like to emphasize on the following points, which form the basis of my presentation today. That is the potential applications of biosensors in food industry, which is ensuring food quality and safety, quality check in dairy industry, freshness check for fruit and vegetables, monitoring of wine quality, and so on. Elaborating them further, first being the food quality and safety. Biosensors check for the desired amount of components of food like content of amino acids, cholesterol, carbohydrates, and so on. Biosensors also determine the shelf life of fatty acids, organic acids, sugars, etc. They also ensure the safety of poultry products. Biosensors for quality check in dairy industry. With reference to this, biosensors are used for the detection of microorganisms, pathogens, allergens, or checking the bacterial load or monitoring the preservation and pasteurization of milk. Let us now move on to the other application, that is biosensors for quality check of fresh fruit and vegetables. Now, if we talk generally, a spoiled food or a stale food can be distinguished from a fresh one by either their taste, appearance, odor, and texture, which are physical parameters. But however, if you want to detect them subjectively, biosensors are used. Biosensors, as I have already talked about, have an immobilized layer of biological material. And in this case, the biological material are enzymes like alcohol peroxidase and alcohol oxidase, and a chromogen is used, which interacts with the analyte, which is basically the food here, and it gives a biological signal, and this biological signal is detected and converted into an electrical signal. This biological signal detects any kind of spoilage or injury, basically, or damage to the fruit or vegetable due to less oxygen amount, maybe in lettuce, cabbage, broccoli, etc. Moving on to the next application, that is monitoring of wine quality. Now here, as it is already mentioned, wine is composed of ethanol, water, sugars, glycerol, and organic acids. And biosensors immobilized layer is basically formed by 3-pyrroloquinoline quinone, that is PQQ, dehydrogenases such as glucose dehydrogenase, alcohol dehydrogenase, and glycerol dehydrogenase. The important enzyme substrates are oxidized, which are glycerol in case of GIDH, glucose for GDH, and ethanol in case of ADH. Now, biosensors can basically detect agents of bad odor and flavor in wines like 2,4,6-trichloroanisole. This brings me to the last application of biosensor, that is antioxidant and heavy metal detection. As we all know that antioxidants, as their name suggests, are those compounds which have the ability to inhibit oxidation in food products. So a type of biosensor, namely amperometric biosensor, can be used to measure the antioxidants in food. Amperometric biosensors are the ones in which there is mensuration of the reaction of analyte with enzyme. Another one is microbial biosensor, and as it is evident from its name, these use genetically engineered microbes and enzymes like oxidase, urease, alkaline phosphatase, etc. These biosensors are used in conjugation with electrical or optical transduction systems. One more biosensor employed in this reference is an enzymatic biosensor. It is basically used to detect the enzyme inhibition caused in water. Enzymes which are used are alkaline phosphatase, acetyl cholinesterase, which are inhibited due to the heavy metal presence like carbamates. Now I would like to address certain advancements made in this field. The prominent one being ECL, that is electroluminescence sensors, which are a conjugation of electrochemistry and luminescence. These are highly sensitive and they show a wide and dynamic range of detection for food safety parameters. Lastly, we have a slide about trending sensors like e-nose and e-tongue. These are quite interesting and they are also useful. These devices are basically having sensors that correspond to the odor and analyze its chemical makeup to identify it. In fact, they are much more sensitive than the human nose. Just like human nose, they basically detect the olfaction, that is the smell sensation. Similarly, e-nose detects the olfaction and sends signal to the program which corresponds to the human brain as it is depicted in the diagram. Now the same goes for e-tongue. This is also a novel approach that is used for taste evaluation of food, and it has a taste assessment array, as we can see here, sensor array, similar to that of human tongue. It tastes soluble food, hence it is used in liquor or beverage industry. So to sum up, we address the potential applications of biosensor devices with respect to the food industry, which may help in ensuring food quality, purity, which are our main concerns, and that too in lesser time and rapidly and more effectively. This advanced approach will definitely accelerate other various aspects of applications in food sector. And this brings me to the end of my presentation. Thank you. References which I refer to for my presentation today. Thank you.
Thank you so much. Very nice presentation. Please, next one.
Hello, sir.
Sir.
Sir,
Yes, sir, please.
next participant is Kamalpreet Kaur. She can't unmute herself, so kindly do it.
Okay, sure.
Yes, sir. It's Kamalpreet Kaur, sir, from Khalsa College. It's already there in the attendee, sir.
Kamalpreet Kaur, please present your paper.
Hello, sir.
Excuse me, sir, आपकी आवाज नहीं आ रही है.
Yes, sir. I think there is some technical issue, sir. That is why I am presenting my next student. That is Harmeet Kaur.
Okay, please, sir, please.
Yeah. Kamal, please unshare this slide. Sir, is it visible?
Yes, very much visible. Please proceed.
A very good afternoon to one and all present here. I am Harmeet Kaur, student of Khalsa College Patiala, and right now I am pursuing BSc Biotechnology Honours. So today I will be presenting a presentation on the topic biotechnological agents used in biowarfare. So here are the contents which we are going to discuss today. So Clostridium botulinum and the Francisella tularemia. These are the both agents which we are going to understand today. So here is a brief description about the agents. The first one is Clostridium botulinum. It is the gram-positive bacteria which forms spores, and the toxic this bacteria produces is neurotoxin botulinum. The other one is the Francisella tularemia. This is a gram-negative bacteria, non-spore forming, and the disease this cause is known as Francisella tularensis, and its other name is rabbit fever. So this slide shows the mode of infection, like how they spread. Clostridium basically spreads through the contaminated food or the untreated water, whereas Francisella tularensis majorly spreads through the bug bites or the bacteria which is present in the air, or majorly through the laboratory exposure. Now we will discuss about the mechanism of the pathogenesis of the Clostridium botulinum. The bacteria enters the body. It tends to produce the neurotoxin botulinum. This toxin then binds to the motor nerve terminal, which basically blocks the release of acetylcholine. So if it is not produced, no muscular contraction will be there, so which will consequently lead to the paralysis. Like here in this diagram, this is the normal mechanism, the first one, where acetylcholine is being released and receptor detect that, and the muscle get the signal to contract or relax. Whereas in the second diagram, the abnormal mechanism, the toxin binds to the nerve terminal, whereas the acetylcholine is not produced, and this leads to stop in the muscle contraction. Now the other one is the Francisella tularemia. So once it enters the body, it infects and multiplies in the macrophage. It enters the cell in the form of phagosome, then it breaks out of phagosome. It replicates multiple of times and then undergoes apoptosis. This leads to swelling in the lymph nodes, and symptoms are like ulceration on the skin. So this slide shows the symptoms of Clostridium botulinum. The first one is the drooping eyelids, and the second one is the blurred vision. And these are the symptoms of Francisella tularemia. These are the ulcer, or this is a leg ulcer. So this slide shows the treatment. First one is Clostridium botulinum. There are basically three types of botulism diseases. First one is foodborne. The bacteria enters through the food. In this case, antitoxin is being administered into the body of the patient. The second one is the infant. This happens in the case of infants. Enema is done here to remove the contaminated food. The third one is the wound botulism. In this, the bacteria enters through the wound, and the majorly antibiotics used are ampicillin, tetracycline, or the penicillin G. Then there is the Francisella tularemia. The majorly antibiotics used are gentamicin, doxycycline, streptomycin. And then here are the miscellaneous points. So in the case of Clostridium botulinum till 2011 we were having the vaccine, but it was discontinued, and now the new vaccine is being under development. The other point is 1 kilogram will be sufficient to kill the entire human population. The fact about Francisella tularemia is that in 1920 to 1930, Francisella tularemia was one of the 7 biological weapons which German and the American corporation had at that time. So I would like to conclude here my presentation. So biowarfare comes under the dark biotechnology. Clostridium botulinum causes the inability to breathe or paralyzed muscles, whereas francisella tularemia leads to the swollen lymph nodes and the ulceration on the skin. So according to my presentation, both the Clostridium botulinum and the francisella tularemia have the ability to become the biowarfare agents. So here are the references, and thank you so much.
Thank you so much. Nice presentation. Please, next one. Please, next one.
So next presentation is my student Sushamjot Kaur, and she's presenting a poster.
Okay, sir.
Good afternoon to one and all present here. It's an honor for me to represent myself in this international conference. I'm Sushamjot Kaur from Khalsa College Patiala, class BSc Honours Biotechnology. I'll be presenting on the topic pathogenesis and treatment approaches for acne vulgaris. Starting with the abstract, I will be discussing about pathogenesis and treatment of acne, followed by mechanism of action of a drug, benzoyl peroxide, and in the end, a novel method for treating acne, which includes lipid nanocarriers. Introducing to acne vulgaris, it's a common chronic inflammatory disease of the skin which affects the pilosebaceous units of the skin and leads to inflammatory or non-inflammatory lesions. Now, the factors leading to acne production are stress. Psychological factors such as stress and anxiety can cause acne. Genetics, hormonal conditions of parents can be a factor. Comedogenic medications such as steroids, lithium, corticosteroids, etc. can cause acne. And smoking in youth can also be a factor. Further, I have talked about the pathogenesis of acne and the events involved in it. Now, if we look at the diagram, first diagram is about a pilosebaceous unit. Second diagram shows the early stages of clogged pore development. Third diagram depicts the full development of the clogged pore, which occurs due to the aggravation by hyperkeratinization and excess sebum production. Fourth diagram represents the occurrence of acne lesion by proliferation of anaerobic bacteria by inflammatory mediators. Inflammatory infiltrates are responsible for severity of acne lesion. Proceeding on to the methods of treatment of acne. These include topical treatments such as retinoids and antibiotics, combinational therapies such as benzoyl peroxide with topical antibiotic or retinoids with a topical antibiotic. Physical treatments include comedone extraction that involves extraction of comedones with the help of certain tools and instruments, and cryoslush therapy in which intense cold conditions are created to freeze or destroy the diseased tissue. Last, treatment of acne, systemic treatment, which includes oral antibiotics and hormonal agents. Moving on, I'll be talking about the mechanism of action of benzoyl peroxide. Here, benzoyl peroxide is converted to benzoic acid, which is then absorbed and metabolized by cysteine in the skin, which releases free radicals that destroy the bacterial cell wall. That will lead to imbalance in osmoregulation, thereby allowing exchange of materials across the cell membrane, hence the organism dies. Further, I'll be talking about the novel method for treatment of acne that includes lipid nanocarriers, which accelerate the onset of drug action. And in the end, I would like to conclude that in this poster, I have talked about the pathogenesis of acne, the mechanism of action of drug benzoyl peroxide, and a novel method to treat acne that includes lipid nanocarriers. So here are the references that I have used
Thank you and have a nice day.
Thank you so much. Next one.
A very good afternoon, sir. I am Swati Sharma from class BSc Biotechnology of Department of Khalsa College, Patiala. I am very glad to be a part of this international conference. Here's my presentation is regarding the immune aspect of the cancer. Here is my content. So what is cancer? Basically, cancer refers to the magnet tumor, a result of the infinite rapid uncontrolled cell proliferation and invasion of the normal surrounding tissue. So depending on the tissue of their origin, there is three types: carcinomas, leukemias, and sarcomas. Carcinomas is in majority and arises from the ectodermal tissue, that is skin or internal organ linings, glands, etc. The most breast and lungs, colons, other cancers are arise from the carcinomas, and there is a leukemias, which is hemopoietic cell of the bone marrow, second most common, which is about 9 to 10% of it. Third one is the sarcomas arise from the mesodermal tissue, which is very in a rare manner, which is 1% at least. Then how magnet transfer from the normal cell to the cancerous cell? It is about the three agents lead to the cancerous cells of the normal, that is carcinogenic chemicals, which is causing the common local damage of the DNA. Examples are the DNA chemicals like DNA alkaline agents or content of tobacco. Second one is ionizing radiation like UV rays, X-rays that causes a chromosomal break. And third one is oncogenic virus can causes the dispersion of the normal genetical and cellular growth of regulation and differentiations. Then these many genes in our which tumor released, these three are involved in a magnetic transformation of tumor. First one is gene promoting, which promote the growth and proliferation of the cancerous cells. Then other is cell inhibiting cell growth, which inhibit that the cancerous cells. And the third one is gene regulating apoptosis, like BCL2 genes, which apoptosis. Third one is the tumor antigens. This antigenic, which is antigenic substance produced in a tumor cells that is triggered in a in a immune response of the host cell. They have no membrane of protein. These have diverse specificity. They act on a particular genes. Based on their uniqueness, there is a two classification of it. First is tumor specific antigen and second is tumor associated antigen. Tumor specific antigens are the unique protein present only on a cancer cells and not on a normal cells. They found in both spontaneous and development by the physical or chemical agents of viral agents. Second one is the tumor associated antigen, which are they are not unique to the tumor cells. They are expressed in a low level or early age of like fetus in the fetus development. They are arises by the increasing the expression of normal cells and by the activation of the embryonic gene due to the carcinogens. My other slide shows that there is a theory, immunosurveillance. It is a theory state that the system patrols the body. Not only they recognize and destroy invading pathogen, but also the host cell that become the cancerous. This included many cells of our immune system like NK cell, natural killer cells, cytotoxic cells. Third one is cell mediated T cells and B cells. T cells and B cells are very much specific for the cancerous cells, and there is dendritic cells which macrophage all the cancerous cells and act on it. Here is the diagram which shows the cell cycle about it and immune response, how our immune response to the cancerous cell. There is first the release of the cancer cells in antigen, which in which our dendritic cell or other cells which engulf and macrophage and represented by the T cells in lymph node, and then they activate all the cell immune response of the cells like macrophage, NK cells, T cells, B cells, and they goes on a blood vessels and infiltrated of the cancer cell by the immune system. Then the T cells recognize the cancer cell, and by the killing their cell and immune response are occurring. These then how the these response of our human cells, but there is also cancer cells on it, but there is a immunotherapy which have main aim of enhancement of the immune response to tumor in a host. There is three approaches to be tried currently. First is immunogenicity, second is passive immunity, and third is cancer vaccine. There is also a monoclonal antibodies and cytokinin which attract all the cells to inhibit and against the cancer cells. There is a conclusion that overall the cancer is caused due to the genetic or environmental factor. Both are involved in the tumor formation. We are talk about the normal cell transformation, how they transform from the normal cell to the cancer cells. There is genes and antigens respond to the cancer cells in our immune cells. There is immunotherapy, which is current area of research to find new treatment modalities. Monoclonal antibodies, adaptive cell transfer, enhancement of the APS in function of the tumor cells are current approach to be explored. Here's my references, and thank you and have a nice day.
Hello. Hello, good afternoon everyone. Myself Sushi Mittal, student of PSC Biotech. Is I audible?
Yes, very much audible.
Okay. Sorry. Hello, good afternoon. Myself Khushi Mittal, student of PSC Biotech Honors, Khalsa College, Patiala, Punjab. I am glad to be part of this international conference. Today, I am going to represent on a silent killing disease, that is a coronary artery disease. So in this representation, we will discuss about brief introduction, its main causes, case study, diagnosis and treatment, developments made by biotechnology, and lastly the conclusion and references. So starting from the introduction, coronary artery disease is a chronic disease and it is the most common type of heart disease. It is the leading cause of death for both men and women in the United States. According to the Center for Disease Control and Prevention, it is the most common type of heart disease in the United States, where it accounts more than 655,000 deaths per year. Someone may first notice that they have it after experiencing angina symptoms like breathing problems, headache, heart attack, but mainly it can cause heart attack without any previous symptoms, and this even go unnoticed, and it is known as a silent heart attack. Next, we will discuss about the main causes. Coronary artery disease happens when the arteries that supply blood to our heart muscles become hardened and narrowed. This happens due to the building up of cholesterol and fatty material called plaques. The collection of plaques is known as atherosclerosis. Due to this, our heart can't get the enough blood or oxygen that it needs. This situation can lead to chest pain and discomfort called angina factors and also even result in heart attack. It also results in the arrhythmias. Arrhythmias are the changes in the normal beating rhythm of our heart. Due to this problem, our heart may not receive enough oxygen-rich blood and physical activity, and this condition is known as leukemia. As we can see in our figures, this is a broad view of our coronary arteries. Mainly our arteries consist of two inner layers, that is tunica media and tunica intima. In the first figure, we can see that our plaques are starting to develop in our artery walls. And in second figure, we can see that plaque mostly covers our tunica intima, the innermost layer, and it results in a very small part of our blood flow or oxygen supply. So next is a case study of a 21-year-old boy. His appearance and ECG result reports are as shown in our chart, and as we can see from here that his blood pressure, pulse rate, oxygen saturation and mild dyslipidemia are way more than normal range. Main reasons for his problem is his 3-year pack smoking history and his family history, which include his maternal aunt and great grandparent. In the combination of his problems like mild dyslipidemia, hypertension, obesity, smoking, and his family history was sufficient to induce severe coronary artery disease at his just 21 years of age. Now, talking about the diagnosis and treatments, as with the help of new technology, we have different methods of instruments to cure. Diagnosis treatments like electrocardiogram, which measures our heart's electrical activity and can assess heart damage. Second one is stress test. This involves walking on a treadmill or paddling a stationary bike in the clinic while your ECG, heart rate and blood pressure are monitored. Third one is chest x-rays. Fourth, blood test. Blood test involves your levels of blood sugar, cholesterol and triglycerides. Fifth is cardiac catheterization, in which a doctor guides a very thin tube called a catheter through blood vessels in your arm or leg to reach your heart. The doctor injects dye through the catheter and uses X-ray videos to see inside your heart. Treatment includes lifestyle changes like quit smoking, learn effective ways to manage your stress. Also the medications like anticoagulants, immunosuppressants, etc. Third one is angioplasty. Angioplasty is used by doctors to open up the blocked artery without opening up chest. In this, doctor threads a thin flexible tube with a balloon through your blood vessels until it reaches the blocked artery. The doctor then inflates the balloon, which stretches the artery open so more of the blood can flow. Next one is coronary artery bypass. Coronary artery bypass graft involves taking a blood vessel from another part of the body, usually the chest, leg or arm, and attaching it to the coronary artery above and below the narrowed area or the blockage. And this is a major operation and it will probably need to be hospitalized for 5 to 6 days. Next one is advancements made by biotechnology. There are various advancements that are made by biotechnology by collaborating with tissue engineering. The first one is stem cells. According to the data presented at American Heart Association's Scientific Session 2021, researchers found stem cell therapy to reduce major cardiac events in individuals with heart failure. Tropic mediators secreted by stem cells improve cardiac function and also reduce the inflammation. Next one is 3D printing and drugs. 3D heart models are made up of flexible materials for use with a mock circulatory system. Such models allow the stimulation of surgical procedures under mock physiological conditions and are therefore potentially very useful to clinical practices. And the last one is nanotechnology. Nanotechnology is now one of the biggest killer of the Western world. It can increase the efficiency of our drugs, systematic delivery to the...
Start, please. Yes, the chief guest has joined actually. Na?
Okay.
Ji, sir.
Lastly, we will talk about the conclusion. So in conclusion, we can say that coronary artery disease develops when coronary arteries develop cholesterol deposits in their walls, and the condition causes blockages in the arteries that feed oxygenated blood to the heart. The arteries get stiffened and inflammation increases. It can be difficult to treat and also cause heart attack or stroke. However, people can take steps to reduce their risk of coronary artery disease by regular exercises, adopting a healthy diet, and avoiding tobacco or cigarettes. People should seek immediate medical attention if they have chest pain, breathlessness, any discomfort known as angina symptoms, as this could indicate a heart attack. In the last decade, consecutive developments have been implemented by biotechnology. By collaborating with tissue engineering in policies like stem cells, 3D printing, nanotechnology, and many more. Regular 6 months full body checkup should be done, as we all know that prevention is better than cure. So here are my references, and that's it from all my side. Thank you for your precious time. Good afternoon, one all present here. I am Kamalpreet from Khalsa College, Patiala. Here I am share my point of view on the topic of Ayurvedic formulation as strategy to cure the diabetic mellitus. Bringing light on the current report of World Health Organization, there is a more than 500 million people who are suffering from the diabetes. That is almost to the double the population that are suffering in 2010, and it becoming the leading cause to having the severe diseases such as kidney, liver damage, and cardiovascular activities, and it becoming the leading cause. It become the leading cause for the death. The fun fact about the diabetes that the having too much sugar in your food isn't the main cause for diabetes. However, this huge increase in this number is resulting from the 21st century lifestyle that include a diet full of sodas and processed food. So basically, the diabetes is a complex metabolic disorder that lead disbalance in the blood glucose level, that either from the insulin insufficiency or the insulin dysfunction. At the broad spectrum, diabetes can be classified into two types. First type, the diabetic patient cannot produce enough insulin, and or the second scenario, that the body isn't able to utilize the insulin that produced by the body. So as I mentioned, that diabetes is a life threatening disease. However, till today there is no such proper medication available for the diabetes. But however, there are the many drugs like metamorphin, sulfonylureas, and OPA4 that can be used for the maintenance of the blood glucose level. But however, these drugs have a major side effects that are more harmful, that can cause weight gain, liver toxicity, and the cardiovascular activities. So here we come to the main point of this discussion, as the diabetes isn't a new disease to mankind. Historical account reveal that the 700 BC is a well-recognized disease in India, and the Ayurvedic remedies are used for the treatment of the diabetes. According to the according to the reports, there are more than 800 plants that have been used that can be used for the treatment of diabetes, as they're having the anti-diabetic potential. In my presentation, I'm trying to cover some plants that have anti-diabetic potential. So firstly, I'm going to start with the most commonly used plant for diabetes is a tulsi.
So the aqueous extract of the plant shows significant reduction in the diabetics patients. And then moving further with a Vijayasar plant that it plants flavonoid extract the marsupium and petrosupium have shown anti-diabetic potential in type 1 diabetes and can lead to production of the insulin from the damaged pancreatic cells. And moving further with the jamun shown an anti-hyperglycemic effect and that can also inhibit the insulin activity from the liver and the kidneys. Then the avla is a traditional plant that used for the therapeutic in the diabetes. That's metabolic extract can also show the anti-inflammatory, anti-mutagenic, anti-carcinogenic, and anti-diabetic activities. And then the major point of this discussion is that the amino acid extracted from the fenugreek, that's a 4-hydroxyleucine that increase stimulation of the glucose from insulin from the isolated cells of the pancreas and also prevent the cell body from the antioxidant activities. And then we move further with the plant, the Trinospora cordiflora.
Stop.
What's sir?
Stop. Try to next participant. So we are. Please next speaker anyone. If next, Siddharth, please call to next presenter.
Thank you, sir.
Mr. Siddharth, are you listening me?
Yes, sir. Yes, sir.
Yeah, call next presenter. Yes.
Good afternoon to one and all. Here is Kashvir Singh from Department of Biotechnology from Khalsa College, Patiala. And today I'm presenting the topic of biofortification, which is novel approach to enrich micronutrients in field crop, which is a very big epidemic. So firstly, I would like to tell that why we need this biofortification. We need biofortification firstly to tackle the upcoming silent epidemics of malnutrition, because it is arising due to population growth, because population is arising at very large rate. Other thing is we want to fulfill our daily needs as well as micronutrient needs, which is essential for holistic development of a body. So we need biofortification to protect citizens from these deficiencies, because as we seen in this picture, malnutrition also risks to death. To explain biofortification, it comprises of two words. First is bios, which means life, and other one is fortificare, which means make strong. So whole meaning is to make the life strong. Biofortification is an approach, or we can say it is a strategy by which we can enrich the micronutrient level in plants. Like nowadays, the deficiency which is mainly occur is iron, zinc, and vitamin A. So by this biofortification method, we can deal with these nutrient deficiencies. And we can enrich generally in the crops like rice, wheat, and maize. To be precise, there are generally three methods for biofortifications. First one is a conventional breeding, and second one is agronomical method, and last one is a genetic modification. In brief, conventional breeding is like in this we crossbreed a parent with as we see in picture. First one is a virus resistant and second one is a high yield crop. So we can crossbreed it and we get a crop with virus resistant as well as high yield. So this is a controlled breeding of crop plants. Second one is agronomical methods. It involves like application of fertilizer, biospray, manure or any other techniques for nutrient enrichment. As we see that there are 4 methods. First one is by fertilizer, second one is by foliar spray, third one is a intercropping and fourth one is using manure. So by this we can use biofortification, and to explain the merits and demerits, it is very easily practiced by farmers. And second thing is it is practical approach. Move on to the genetic engineering, which is a last method. But in this method, we can insert a gene or we can clone a gene. It is done in various ways. Like firstly, we can clone a gene with the specific quality. Second, we can design a gene in specific way that it can give us better yield. Third one is that we can locate the particular trait in a gene to make it more efficient. At last, to conclude it, overall biofortification is a perfect strategy, because nowadays population is increasing at a very high pace. So it is a good way to control the micronutrient deficiencies. Conventional breeding and agronomical methods are practical, but they are not efficient as compared to the genetic engineering, which is very efficient, as it is. By this we can decrease the use of pesticides, as well as we can also get better yield without less toxicity. So here are the references which I use to present this topic. Thank you and have a nice day.
Thank you so much. And next participant, that is the last participant.
Sir, I think all the presentations from Khalsa Patiala are done. The last one was Shusham Jyot Kaur. She already presented.
Okay.
Thank you so much. Thank you, sir.
I mean, every presentation has been successfully included?
Yes, sir. Yes, sir.
Okay. So
now I will send you the marks of all the presentation, and all the jury members mark I will send you. And in this time now, I hope a summary of the 3 days conference.
Yeah. So very good afternoon to you all. We are here right now in position to conclude our 3 days event, International Congress on Recent Trends in Animal Sciences, Life Sciences and Biotechnology. And we are warmly welcome to our chief guest, Professor Anil K. Sharma sir, Head Department of Biotechnology. And in his gracious presence, we will follow towards the valedictory session. So over to Siddharth. Please move ahead according to the proceeding cost for the overview and summary presentation of 3 days event. So over to Mr. Siddharth.
Thank you so much, sir. Now I would like to invite our co-organizing secretary, Ms. Bhavna Paliwal from Banasali Vidyapit Outreach. Can you conclude this session with 3 days summaries? Please, Ms. Bhavna Paliwal.
Hello, am I audible?
Yes, very much audible.
Good afternoon, everybody. This is Bhavna Paliwal, and co-organizer and co-reviewer of this conference, the 6th annual meet of the International Association of Zoologists and the 3rd International Conference on Recent Trend in Life Science and Biotechnology, organized by International Association of Zoologists and Siddhartha Group of Education.